इब्सा एक बार यूट्यूब देखो ना, आ रहा है क्या? should come now. आपको भी स्ट्रीम किया आएगा? हाँ यही है। ठीक है, फेसबुक करके रखता हूँ। हाँ, pretty cool for you. Yeah. Okay. Is it the same thing for you? No, okay, right? Yeah. Can you bit what I don't know. Yeah, love your. Yeah, I love your. It's like. Yeah, that might be easy. It's really cool. I get love your. Yeah. अगर खाना खाना है तो ये ब्लॉक है मतलब पूरा पेट को खराब करेगा. So you had your lunch? Yeah. Then you can have it. Not me now. I'll ask if you want. Okay, don't worry.
So we'll get started in 1 minute, please. Yeah.
Lunch is done?
Yes, sir. Have you taken your lunch?
Yeah. I've taken this character. Do you want it?
No, it's fine, sir.
Okay, thank you. No, we have a problem. मुझे कोई इंक्रीज करो. हाँ? ये वाला बस इसको वही केवल बोल नहीं मुझे उसने पूरा मारा कर दिया. सर आई एम नॉट शेयरिंग ईमेल इज मेड. ओके ओके. चलो फिक्स करता हूँ. हाँ. Because everybody is asking actually. No, we अभी हमको टाइम है ना? हाँ वो कब है? 10 का सेशन. Yeah, after question session we do it ना. So I'll fix it before.
So Hiten Subbu, you can start the recording, please. Yeah.
Yes, I'm starting.
Yeah.
Okay, Prash, the recording is started. Yeah, the next chair is Prashant Suravjanala. He will take care of the next session. Thank you, Prash.
Thank you, Kiran. For this wonderful hosting. Yeah. So yesterday, I had a great pleasure in inviting Gareth Price. But Kiran today this morning has wonderfully introduced Gareth, who needs no introduction. He is really kind enough, to accommodate Dave Clement's talk as well. So today it's going to be his, and this is going to take us, to the world of this NGS, more precisely into metagenomics, epigenetics, transcriptomics, and much more. So, what I would probably request the audience over here is, to be more interactive. Please ask questions. This is probably, one of the kind of a rare opportunity that you tend to interact. Gareth is coming absolutely in a different time zone. It's 6 p.m. for him in Australia, and most of you are in India. But you can probably set a precedent, for this Galaxy workshop, by interacting with the speaker, and he's none other than Gareth Price. Yeah, please, thank you so much, Gareth. Yeah.
My pleasure. Thank you. And thank you, everyone. I hope you had a good morning so far and a good lunch break. As Prash just indicated, it's coming into our evening, but that's not going to stop me in any way. And I certainly echo the comments just then that feel free to interrupt as you can if there's something you want me to explore deeper. More familiar with Google Meet now, so I will have the Google chat up on my screen. So please do interact. Otherwise, over the next hour and a half, there will be time for questions near the end. But I'm going to give you a little bit more of an in-depth view onto Galaxy and how to use it and some of the features. Now, I have the luxury, I guess, of being able to double present today after I presented on Dave Clement's behalf and Galaxy project this morning. So I've tried to keep the two slide decks slightly independent of each other. However, I also recognise that if there are some people joining that were earlier, there will be 1 or 2 slides to cover things off. So where were we at the start of the day? Well, I tried to go with the space pun that is galaxy. So this one is from the stars to the Earth, and this previous presentation was all about the community, the coders, the tool wrappers, the training content creators, and the trainers, and really to re-emphasize that the community we're trying to build Galaxy for and operate Galaxy for is the scientists that need it and the scientists that ask for it and use it in their publications. So what I want to emphasize in this session is a little bit more about the Galaxy Training Network, ideally a little bit about batching data into workflows and workflow reports. The workflow reports being one of the newer features of Galaxy, but one that again, I think as an individual, quite attractive, but also as a mechanism for sharing data, very attractive as well. We have a raised hand, so I'm not sure if we have a question already.
No, Garrett. It's basically just people are entering, so the sounds are coming up.
Okay, not a problem. Thank you so much. Yep, I'll do feel free to interrupt if there's a burning question. Thanks. So, as a reminder, and this time I've shown Galaxy Australia. The tagline, as said before, it's a web accessible platform that allows you to conduct accessible, reproducible, and transparent computational biological research. Now we're going to dive into how it does that. So, as a reminder, the tools on the left, your history on the right, that contains your
Garrett,
data.
sorry to interrupt. Are you already sharing the screen? Because we don't.
Oh, there, that's the issue. Apologies, I thought that was being shared. Yes, maybe that is why I got a bit of thank you. Share screen one. There we go. Bit of a rookie mistake. Apologies for that.
No problem. Maybe you could go for a couple of things if you missed. Yeah.
Yes. So I've just skipped over the title slide. So, and I'll just see if I can't shrink that bar down. There it goes. So what I'm indicating before, and thank you for that catch, is that we have previously talked about who the Galaxy community is as people, the people that deploy the service. But really what I want to emphasize in this particular session is how to get the best out of Galaxy and certain features that I think make Galaxy the approachable platform it is. So we're going to focus on the Galaxy Training Network, some import rules to data, workflows, which I'm a huge fan of, and then workflow reports as well. And yes, thank you. I was up to here to show the screen. So Galaxy, this is what it looks like. Tools on the left-hand side, the history on the right-hand side, and this middle window of interaction. The spaces we're going to work in a bit over this session or highlight are the workflow section up the top menu, maybe a bit of a feature on this little academic hat here. And given the number of questions in the earlier session today about storage and quota, which are very reasonable questions for people to ask, at the very top right hand of your screen, you will have a percentage of your allocation that you're using up, and that is a great place to keep an eye on your quota, potentially purge old data you don't need, purge analyses that worked or didn't work to keep it under 100%. And you can see when I took this screen grab of my Galaxy instance a while ago, I was reaching the top of my threshold. Yep. So what is, we talked today and we're going to continue to talk about Galaxy as a tool repository. But I guess I want to switch focus a little. For me, Galaxy is about running workflows. So individual tools are great, but you could imagine if I want to run one tool, I don't really want to sit there watching my screen update until that tool's finished, execute the next tool, the following tool. It's effectively watching a kettle boil, or in this particular case on the screen here, about 30 kettles boil one after another. So workflows are where Galaxy really gets its power. There are other workflow languages out there, of which popular is SnakeMake and Nextflow, but they do require, in general, some command line experience. In Galaxy, you can visually chain your workflows together, and we'll look at that as an exercise shortly. Execute that workflow. Importantly, inside the workflow, you also get version control. And this screenshot of mine is a little old, and we're going to have a look at closer detail live at the India Galaxy instance shortly. You now get best practice prompts for, is the workflow annotated? Is it authorized? Have you given it the information to give this workflow longevity? And we will get into some live demonstrations of Galaxy shortly, but one of the challenges we do know that exists in Galaxy is the increasing demands on bringing your data into the service and streamlining that as much as possible. So I've listed up the top a number of features, one of which I need to update, but I'd just like to sit through them fairly quickly. So local file. This is a file on your computer that you're going to interact through your Chrome, Safari, Firefox window and upload into the service. There's absolutely nothing wrong with that process. However, it's bound to your own computer, so you can't turn your computer off, you can't unplug it and walk away. And it's mediated by your computer to web connection. So if that drops out for any reason and you're uploading a very large file, then that connection's severed. So again, it is a very useful option, but increasingly less demanded because of the increased file size. There is an option on different Galaxy servers, and I believe I saw this is enabled on India.use Galaxy, and that is File Transfer Protocol, or FTP, in which case you will mediate the interaction between your computer and FTP server. There's a temporary hosting site on Galaxy and then import from FTP into Galaxy. And that works really well. So I do encourage people to use that if you have data. It's not the fastest route in, but it does work well. Pasted data. This is literally typing into the upload box what you want to go in, and it's very useful for things like small annotation files. I'll speed up a bit. So URL, this is essentially what you're looking at down on this scrolling GIF at the bottom here. This is actually the rule-based URL, but you can see in the last column here, column D, the last column before it scrolls, this is a URL. So any URL that is publicly exposed can be ingested into Galaxy. And that's quite an efficient way, because then you're doing server-to-server data ingest into the service, but also that URL needs to be publicly available. So you do need to consider if you can publicly expose your data even for a short period. If you can, then the rule-based imports are fantastic, because you can bring in all the metadata associated with those files. You can auto-build a collection of files on import. So I think we're going to see this feature used a lot more with data sets in the future. And the final version here, which is in the latest Galaxy, is actually no longer Cloud Store, which is an Australian-only service, but rather remote data stores. So if there's a publicly available data store or a private one that you can configure, like a Dropbox account, then you can link your account to your Galaxy instance and mediate data transfer this way. Why I'm belaboring all these descriptions is because data movement and data custodianship is becoming increasingly relevant in how we see people interact with Galaxy. And I would strongly encourage you, if you can move your data from the, say, sequencing facility it's been produced at into a cloud repository like Dropbox or AWS and then into Galaxy, that is going to be the smoothest transition into the service. So the next feature, and we're going to explore this one a little bit deeper, is quite a fantastic feature actually. actually in this one that started on Galaxy Europe, and thank them for thinking of this idea. So by observing in a de-identified manner the usage pattern of Galaxy Europe over a number of months and collecting information about how users interact with the tool and the tool that they're most likely to use after an initial tool, like RNA star in this case, they were able to build a predictive algorithm for downstream tools. What this means is inside a workflow, but also inside a simple tool execution, you can now get some essentially community and we hope best practice hints of how to chain one tool to another tool to another tool to produce a biological outcome. So this is really quite fantastic for people that are maybe trying to understand how to use the output of a particular tool that's been recommended to them or they've read about, to see how to use it within a workflow. So, and this is available on other galaxies, but specifically it is on Galaxy Europe and therefore India. And one of the next features we're going to talk about through part of the live demo is quite a new one, and one that I'm particularly keen on myself. And this is using metadata tags from your Galaxy workflow, such as Galaxy History Dataset ID, specifying a Markdown language report and auto-generating an not an interactive report, a HTML report, which you can share with others about your data. So the reason I guess I'm so excited about this is because in my other role, apart from Service Manager at Galaxy Australia, I run the bioinformatics group at QSIF Bioinformatics, and we're doing a lot of production level bioinformatics for researchers. And one of the points that we often reach in our journey is generating the results, but then trying to turn those results into an easily digestible and hopefully visual report. But nonetheless, we're a core facility. You can also imagine anyone actually doing this, someone who routinely puts through 12 samples a time once a week and just needs to see the same style of report or summarization of their work quickly at the end before they proceed to more complicated analyses. You can see students doing this to present to their supervisors or any other combination of examples. The take-home message here is that you can cherry pick from the left-hand side elements of your workflow output that you would like to be presented succinctly in a report and have that auto-generated at the end of your workflow journey, and then share that or have that as digestible information. So we're going to definitely look at that over the next hour. So where do we get all this information? And I realise some of this is a little redundant, but please bear with me. Now, I am citing Galaxy Australia here. The mechanism is slightly different on Europe, but the process behind the scene is the same. So huge number of thousands of tools available. They're on your Galaxy instance, but what happens if that one particular tool you need isn't there? Well, by isn't there, where do you go find it? So the first place to go find a tool is to go to the Galaxy Toolshed as listed here, or simply Google Galaxy Project Toolshed and you'll find this link. Every few months, the total number of tools is updated, and as long as I've been involved with Galaxy, this jumps up about 1,000, every second half of a year. If there's a tool here on Galaxy Australia, for example, you simply say, I would like this tool. I found it on the toolshed. This is the toolshed ID, and we will make our best efforts to enable it for you. And Galaxy Europe the same. References. We talked about references earlier today, so I want to again mention them here. You can discover what references are hosted on Galaxy at datacache at galaxyproject.org. The subtlety of these is any individual reference, and we could think of the human genome build 38, the mouse genome build 9, Drosophila, bovine, any example genome you want to discover. having the genome hosted on Galaxy actually isn't simply enough. Many tools need to index that genome for rapid lookup. Otherwise, your tools would take hours and hours and hours. So when a genome is hosted on Galaxy, it's actually pre-computed for the available tools on that service, and that really speeds up your analysis. So again, if you discover a genome on Galaxy, then it's good to go, essentially. If you discover that your genome of interest is not on Galaxy, then again, I encourage you to write to that Galaxy service and ask for them to install it. In Australia, we do host a few local genomes, and we're looking to push them up to the global repository soon. And I think we looked at this in the last talk, but again, just to emphasize, all this sharing is managed via this CERN virtual machine file system, or CVMS, where reference data and tools are shared between the Galaxy servers. And in fact, what's really missing on this map is any other Galaxy server that wants to make use of this shared repository just needs to link to it, and they get all that shared infrastructure. Now, one of the things I do like to emphasize for Galaxy, and something that people may not be aware of, is that Galaxy is exceedingly transparent. If you want to know how a tool has been run, and I will show this slide shortly, there's an information button. It shows you all the tool settings. Depending on your level of authority on Galaxy India, I will be coming in as a user. On Galaxy Australia, I see my jobs as an admin account. But either way, we give you the entire command line syntax that is related to this job. Now, this is a little bit of a freebie and may not wow many people. If you're looking to take the learnings that you have in Galaxy and translate them to bioinformatics experience at command line, I think this is a really fantastic way to see the syntax of a tool. See where features like grep and echo are, where the exits are, where the settings for the tool are. All this allows you to reconstruct this argument yourself at command line. So again, I'd encourage people to look at that if they're interested. And by the same extension, one of the things that we all know we need to do in our research is appropriately cite primary publications. So Galaxy has the opportunity under your history menu to extract all the primary citations for the tools that you used in your analysis. I find that this is quite an amazing and rapid way to immediately get to the core of the activity you did. I generally promote this to students, but really it's applicable to everyone that's writing up their analyses. So before we switch into it, I think a little bit of an explanation of what happens, say, under the hood of Galaxy and how data's moved around. And really, I don't want to belabor this very much because it is A, unique to each Galaxy service, and B, it's essentially invisible to the user. And that's by design. Galaxy is there to deconvolute your experience, to, as the original talk and quotations were, is to hide the computational plumbing from you. So in this particular case, in Australia, the web interface that you interact with is hosted here in Pawsey on the western coast of Australia. But any of your data could be sent to machines east coast in Brisbane, where I live and work, to our national compute infrastructure in our capital city, and to Melbourne in Victoria down here. And all the decisions about where to send this data is based on that combination of tool and data set size and most appropriate infrastructure for your samples to run at. Oh, sorry, your analyses to run. So you shouldn't be aware of any of this. Europe and America make the same decisions. The point of this is if you curious about how your tool is resourced, if it's coming back to you slowly, if it's failing and it's failing because of a lack of resources, then do contact your service and ask about this, because there are always opportunities to find the best fit location for your data. Now, we've talked about this a little bit already, but we're going to make use of this, or at least you're going to watch me demo some of this shortly. So the Galaxy Training Network is something I really struggle to talk up, can't talk up enough. The material in here is fantastic. The range of topics grows, it feels like daily, and the depth of them is really complete. By walking through one of these tutorials yourself, you'll be well placed at the end with an understanding of the biology, the tools to use, some of the settings and subtleties around operating those tools, and be well placed to utilize what you're learnings on your own data. So I want to work with this as we go forward. We've looked at this already. So yes, multiple language support, persistent public data, and multiple servers that run and so on. So with that in mind, I did want to touch on a number of the topics in my fairly grandiose title before we zoom in on one topic. If you remember the title of the talk, I promised NGS, epigenetics, metagenomics, transcriptomics, and much more. Well, I did decide by presenter prerogative that NGS really covers all of these. There are some basic tools like FastQC for looking at the quality of your data, but probably don't deserve a huge amount of time more than that. So what I've done is extracted out of the training portal site here the number of topics associated with each of these domains. So for those of you that are very sharp-eyed, the previous slide had 6 topics associated with epigenetics, and we have 8 here. The difference being that each of these topics has an introduction slide deck to just ground you in the style of analysis that is operating there. So there are 6 tutorials from ATAC-seq to methylation structure, inactive X, high C, binding sites, so more essentially ChIP-seq, and a bit of ChIP-seq on one of the beta arrays from Illumina. Now, each of these tutorials will have workflows and test data available to them. So if this is something of interest to you, one of these topics, then please jump on in and have a look. By ongoing, we're going into the metagenomics section, and the one tutorial that I'm going to demonstrate live shortly is this one here, 16 microbial analysis with Mother, which is a suite of tools. And short and extended is running every single tool itself, short making use of workflows. And I really do want to talk up the workflows, so we're going to have a look at this short tutorial here. And transcriptomics, not surprisingly, fills a screen. It's an exceedingly popular topic that has been for a very long time, and there's depth of analysis options you can do here. To the point, to me, where this almost looks intimidating, it's not supposed to be. So we have that default introduction to essentially what is RNA-seq analyses. The main 3 here are turning your reads into count tables, your counts into genes, and hopefully your genes into some form of pathway analysis. A rapid expansion is occurring in training material around single-cell RNA-seq. And again, if this is something you're interested in, 1, please get in and have a look at these. 2, feedback is most welcome on these tutorials, as they seem to be changing all the time, but in a good way, because it's such an evolving field. And visualization and a few other notable tutorials like gene ontology enrichment, which are often popular ones for people to do. So really, I would like to spend some time, and I imagine this is where we might get some more interrupted time, walking through one of these tutorials. As I indicated, it is the Mother Short tutorial, and just so that you can see I'm doing this real, we are going to do it on the indiauseGalaxy.eu service. I will stop my presentation there and give a little bit more of an orientation on what we're looking at in our Galaxy experience. I understand for some people this might be your first time, so I will go through various elements of Galaxy. It's a little bit of a tour de force through all the things that one can possibly do in here. I can't cover everything because of the amount of time we have, but I hope to give you a good, understanding of Galaxy by the time we finish this session. So for me, I'm quite familiar with the Galaxy interface, but I'll go through it a little bit. And again, for those of you that are aware of all of these features, I'm sure it will seem basic, but hopefully we're getting somewhere. So again, said it many a time, here are all the tools on the left-hand side of the screen. Each of these menus opens up to more tools. And one of the more interesting things that one can do is you don't need to remember where your tool is. You can simply start typing in the name of the tool, and this will come up on the list. And if you know the tool in question, of course, then that's the easiest way to get to it. In the middle window, we're now presented with this tool. It's versioned. So again, if you're particularly interested in how it's working, you can always keep a good eye on the version numbers. Have somewhat picked a tool at random. We're going to talk about this a bit more. Specify your inputs. You have two FASTQ files, forward and reverse. You have them as a pair, or you have multiple pairs, and we'll look at all three options. In two simple files, I simply need to pick my forward pair and pick my reverse. If I'm in a collection, I just need to pick the collection. And in fact, there's only one collection in my history. And this is something that Galaxy does really well. And I think it's one of the things that enables people to use it quite seamlessly, is each of these three conditions, Galaxy specifies a file type. In this case, it's a FASTQ file. In this particular case, it's a paired collection list, which means if you do not have one of these in your history, as in this case here, it's not going to show up, which means really a lot of the simple mistakes, possibly naive mistakes that we see people making are often caused by inappropriate selection of data for the tool. And Galaxy can really help that by basically helping direct you to the style of data you want. The next most often commented element of Galaxy is about all the settings for a particular tool. And this is both a blessing and a curse. So our settings are here. We have an alignment strategy. We have options for trimming, matching, and we have the penalties associated with mismatching. Now you'll see that each one of these options is pre-populated. Now it is pre-populated, and this is the blessing and the curse. It is pre-populated with default settings, and these default settings come from a number of sources. So the first one they come from is the person that wrapped the tool. They are going to be the primary author of the settings you see in the tool. For the person that wrapped the tool, they should, and I say should, not will, should have arrived at these particular settings by going back to the primary research associated with this tool or the primary publication and taking the author's recommendations for most appropriate defaults. I feel like I'm belaboring this point. So my punchline here is that often users will ask us, one, how do I know to change these, 2, what to change them to, and are the defaults appropriate? Well, they're really the third one may be the hardest question to answer. Are the default settings appropriate or not? I'm going to say yes, they are appropriate to allow you to run the tool and get a result. Then you can start to question how appropriate the defaults are by looking at the quality of your result and iterating through reanalysis if needed. The other answer to this is buried down the bottom, and that is that this particular tool comes from a suite called Mother. There's absolutely no reason that you can't go and look up Mother by primary publication and read the paper and see what the authors of this tool recommend for the settings. So, and this particular one has a wiki page as well. So there's a rich level of support built into Galaxy to allow you to make your biological decisions or your bioinformatics decisions. And I think that's just really my point here is please use the defaults, but be aware that you can always modify them after the first experience. What do I mean by that? And again, like I say, we're heading on a journey, but it is going to feel like a path we take. So this is the file, this is the tool make contigs. In the right-hand side of my screen, starting at the bottom, I have my raw data, and moving up, I have results. These are pre-computed because I didn't necessarily want to wait for everything we're doing today to happen live. But here is a pre-analyzed make contigs file. So let's have a look at this one to emphasize about Galaxy's reproducibility. So I can click on this file and I am given what's called a data preview. So I'm given some information. I imagine this is not particularly easy to see on many of your screens. So for starters, at the bottom of Galaxy are 3 little dashed lines. I can grab this, drag this out, and at least try to format this part of my screen to make it a bit more readable. I can do the same on the other side, and obviously the middle of my screen starts to look quite ridiculous. So we won't be doing that. And I'll also, because I've been thinking about it, just make my screen a little bigger for everyone just to make sure you can really see all the text. So we have this particular file, made contigs. I have a something, 150,000 sequences of a particular format, fast day. I can visualize this data for view data. I'll just shrink my screen back. View data by the little eye icon will load up the data in this middle panel. And a couple of things happen. First of all, it informs me, and this is the same for all Galaxies, that if the file is over 1 megabyte in size, then only the first megabyte is shown. You can show all, but please be aware, like any show all functionality, if this is a very large file, it will take forever to download and cache in your computer. And probably, I'm going to be honest, for very little value, because after I scroll through some number of lines here, there's very little information I'm getting as the user out of inspecting this file line by line. But I do want to inspect the file, and I also want to understand the tool, because that's the purpose part of the work. Incidentally, I can rename the file at any point by editing its attributes here, and I can delete the file if I so choose to. At this point, I'm not going to. A bit of the file I wanted to get to and show you is I can download it. So anything you do in Galaxy, you can extract out to your local computer, which is of course going to be useful if you want to save that for a later time. You can copy a link to this file and share that link if you're wishing for someone to download it. But the buttons I want to make most emphasis on here are the view details and rerun this job. So first of all, view details. here is a slew of information about how this tool was run. So first of all, some fair basics. Pardon me for a minute. Sorry, interrupted by pets. Some information about the data set. Most of this is useful to your Galaxy administrator and not to yourself. What is useful is a complete list of the tool parameters, the command line that was used. So there's the command line used to execute this tool, and it's quite long because we did it on a number of files. Any standard output, any error, if the file throws an error, and other elements about the file. You do actually get some job metrics, which is kind of cool. This depends on the server you're on. So we can see the amount of time it took to run, the amount of memory it used or was allocated. So again, for those of you that are particularly interested or concerned about the volume of data you're adding and are you going to overwhelm a tool, then I encourage you to look at the details to get in and explore that. You can also run the tool subset of the data and increase it up with time. And a little bit of a throwaway, but of course something that is always useful to note is how much would have this job cost to run on commercial cloud infrastructure. So per job to be very small, but you'd be surprised how quickly it adds up. Okay, the final part of tools before we sort of get back into the theme of this session is this rerun function. One of my most favored features in Galaxy. So we talk about Galaxy being a reproducible platform. I think the rerun tool is one of the great places to show it. So by clicking on this tool, I brought up the tool, but I brought it up exactly how it was used before. So I can see the input style, the input file, and I can see all the settings. Now they have been left at default, but what's the beauty of the rerun is I could now change this to -2, execute this job, I can rerun it again here, change it to -3, run this job, and keep on iterating through settings that I'm curious about changing. So when you're presented with a full screen of settings, instead of having to go through and say, oh, last time did I call this 4, last time I gave this an atrocious -10, you just rerun the tool, and rerun as you need to do that reproducible but iterative work. So I think I probably made at that point enough, but the rerun is a fantastic feature in the Galaxy. So I was talking about tools. I then went into tools and a little bit into histories, some cosmetics around about how to use Galaxy. Before we go into the tutorial, I'll give you one more particular piece of information and then we'll see how all this comes together. So Galaxy exists with multiple histories. It's very easy to create a new history. You just click the plus button, you give it a name, and hit return, and away you go. You have an empty history ready to upload data. We talked about uploads before, so I could do data from my primary computer. Can't see that because it opened on my secondary screen. I can choose remote files, and these are all these publicly available data repositories, including the COVID-19 repository. So that's well worth a look, as asked earlier today. But it can contain Dropbox on cloud. So again, if you have the opportunity to configure these for yourself, then you can bring data in from that source. And paste and fetch for URLs. I'm going to navigate now to multiple histories, view all histories. And this is where you have everything that you have conducted on this particular Galaxy service. For me, this goes off and off and off as the various things that I've done at Galaxy Europe. And again, that's worth bearing in mind. Yes, I do work at Galaxy Europe as well as Galaxy Australia. I use Galaxy Europe to explore what my colleagues are doing, to be able to talk to people at Galaxy Europe about their experiences. So there are good reasons why we use a different service. It's very simple to just switch to this history. But before I do that, I think one of the fascinating parts of Galaxy is I can build this new history over here by simply grabbing a file and dragging it over. And it is now copied to this history. Can take a moment to refresh. There it is there. And now I have a history ready to use with my raw data. I am going to switch to this one. Right, so I have just refreshed by hitting Galaxy India and come back to our landing page. Our history options at the right hand side of the screen. I just need to zoom out for a minute so you can see them all. There's an awful lot you can do with your history. You can do fairly obvious things like share it. So you can do this with a colleague, share with a user. That user will need to have a pre-existing Galaxy ID for you to be able to share it with them. So you cannot share this just with a random person. You will need to encourage someone to have a Galaxy account for you to share. So it's internal to Galaxy, but once they have their copy, they have their copy and can view it, do what they want with it. You can publish it, and by publishing it, this means adding it effectively to the shared data section of Galaxy. We're going to talk about some amazing features like extract workflow, but not at the start, not right now. Some cosmetic features, and right down the bottom, two that are quite great to use. Expel tool excitation. This will, with time, bring up the citation list, and there it is there. So that's all the tools we have used throughout making this particular history. And this one here that I'm not going to click on just yet, but a great way to condense down into a zip file or a compressed file your entire history, making it available for you to download and take offline as a copy. So that is an awfully quick whirlwind through some of the Galaxy features. Now I'd like to show you how we couple them together to do a body of work. So I'm actually going to switch back to my empty history at this point, which is this one here. I'm going to perform a few steps, and these steps would be very typical of beginning a Galaxy journey or a data analytical journey. And then we're going to come back to the pre-computed history, just so we don't have to watch computers churn. So for myself, I'm just going to delete this file because I essentially want a clean history. I could rename another history, but also want to show you what deletion does. So deleting pushes it into the background. So I have one deleted and 38 hidden files. In fact, they're all now deleted, which is good. They can be rescued. So you can unhide or permanently delete files. There's no reason why I can't just go purge deleted files. And thankfully I get a warning. So that's great because I might have just accidentally deleted my entire PhD's worth of data, which would be unfortunate. But I also get that they're in here. And this is a great thing, that a Galaxy can be sometimes insidious on how much it remembers, but it will remember that I did have data in here. So let's now switch gear a little and head over to the Galaxy Training Network. So this is the Training at Galaxy project. Just since I've got it open, it's here now in the chat. Let's have a bit of a deeper dive into what we can do here and how we can interact with it in our Galaxy instance. I'm going to show you what I might call the old-fashioned way, which is to have two open windows. I realize that you're going to watch me between screens a great deal. There is an opportunity to see this as a pop-up inside Galaxy. I think this can be quite useful, but I find when I'm delivering training or webinars, I do like to have two open screens. So just semantics. So we are looking at metagenomics. Can I show annotation methods of FASTQ files in Galaxy servers? I'm not entirely sure by annotation, do you mean turning it into a protein sequence, checking it against the database, or maybe more just curation of the quality of FASTQs and pathway analysis? Yep. So these are all great questions, and we'll definitely have time to look at them, but I'll step through this first part to get you a look at how Galaxy works. Pathway analysis, incidentally, is one of our more popular requests. Unfortunately, pardon me for a minute. Yes, it is dinner time in our household for the pets, so they are interrupting this call. Apologies for that. So we'll get to those particular questions shortly because I think they're very important. So the Galaxy Training Network. We've talked about this layout, so let's have a look at a particular tutorial. So we're going to have a look at the 16S microbial analysis short. It does take 2 hours. We're not going to attempt to do all this because really I want to show it at a high level. What I really want to emphasize is do get in and use these. You get all your data sets, you get your workflows, and everything else you really need. And you get some fantastic biology. So in this case, we're getting a good description of what it means to be looking at 16S sequencing, the variable parts of that genome, the primary paper that's been cited, and what it meant for the data that you're going to analyse, and then the schema of that. So I'm not going to talk in detail about that right now because I hope with time, if this is your particular passion, you'll come back and look at it, but I wanted to give this a look-see. And you're absolutely right, Prash. She is a very demanding old lady, that cat. She's 17 years old, so dinner time is a routine for her. That is a poor parent of me to not have done that prior to this session. So, importing data. I'm going to encourage, if anyone is following along with this, don't feel obliged to bring all this data in at once. And in fact, I'm not going to bring it all in at once because it's unnecessary. I'm not going to go through the entire tutorial. What I really want to emphasize is that we're bringing in data from a public repository, Zenodo. I'm only going to grab the first few web links, and I'm going to copy those into memory. At my Galaxy instance now, I'm going to upload data and go to paste and fetch data, in which case I can simply drop in that list of URLs, and I don't need to do anything else actually. Things I will emphasize for those of you into the Galaxy space. Galaxy supports an awful large number of file types, more than I care to think about and more than I care to worry about here. I would strongly recommend you let Galaxy auto-detect the file type rather than specify it yourself. And for that reason is because if I type FASTQ in here are all the different styles of FASTQ data is supported by Galaxy. You're not going to break your analysis too badly by selecting the wrong file type, but I think it's rather just a little bit easier to get Galaxy to pick it for you. You can always undo it later if it's wrong. I'm going to hit start in a minute. This will get loaded into the system. The history will populate with these files, and the transaction, however, is no longer dependent on my computer. So I can shut this down, I can go away, I can wait. This is now a transaction between Zenodo's online repository and the Galaxy Europe server. Now, these are currently sitting queued. waiting for available resources to run. If I'm particularly interested, I can refresh my history, and I can keep doing that till they come up to where they are now, which is running. And by running, this means that the communication between those two servers is happening and that data is being downloaded into my Galaxy history. When it arrives, skipping over a few other things here, we're being asked to turn this into a collection, and this is something I would like to show you how to utilize, because again, it's all about making your work efficient and reproducible, and collections are a wonderful way of doing this. So my files have gone green. I, particularly myself, but hope I can impart this onto other people, always like to have a good look at any data that comes into Galaxy, particularly primary data. I've just downloaded this from a URL. I don't necessarily know that URL or the site, so a visual inspection of the data is a great idea. Nonetheless, this looks like FastQ data, and I'm happy to move on. I would potentially look at all of these. I could look at file sizes to see how uniform they are across the data set, but there's small tasks to do. I'm just going to collapse those. So nonetheless, I have 8 files here, and you could imagine I might have 100 files or 200 files. And if the first tool I want to use is called Make Contigs, which is the first tool I want to use, then now you can see my history is populated. It's found the first file that conforms to a FastQ, and it's made it available to me. So first of all, I have to pick the pair. Then, if I want multiple data sets, I now need to go through and click the pairs over and over. So maybe this is an achievable task to look at the files and configure them, but it could be very easy for me to make this kind of mistake if I have 50 or 100 on my list and inappropriately pair my files together. So we really want to make a basic, clear understanding of how to handle the data. And this is one of the things that Galaxy does really, really well to my mind. So I've just enabled a particular operation over here called operation on multiple data sets. I'm going to select all my files. All of this is well described on the training network, by the way. It's all described here. So it is described in other tutorials. So I'm just showing you off firsthand. And I'm going to build a list of data set pairs. So what I'm doing is now telling Galaxy, here's a list of my files, and here's where to find the name of the forward read and the name of the reverse read. So Galaxy operates on essentially a traffic light system. Green, everything's happy. Orange, stuff is running. Red, we're not happy. So we've got red all over our screen. Not particularly a great place to start, okay? I did expect this red, so I'm talking about it. So what it's saying is it cannot find any forward reads and it cannot find any reverse reads. It is because it's looking for these reads by _1 and _2. Unfortunately, our reads yes, so I'm going to cancel this because, I'm not going to cancel, I'll just zoom out and then you'll be able to see. Yep, cancel. So what have I done? So actually, my reads are now _read1 and _read2. So they're not R1, R_1 and _2. I'm going to add an R into here. And as soon as I do, you can see my list is now populated with all my forward reads. If I add one here, I'm now populated with all my reverse reads. And Galaxy's attempted to line these up. So I can now auto-pair these files. They're paired, the suffix is truncated off. I'm given 4 files. I can give this a collection, and I'm going to call it INBIX_metagenomics. Create collection. Yeah. Hiding. So what is actually done is now made a list of pairs by sample, and within each of those is forward and reverse. Which now allows me to come back to this primary tool here, make contigs, rather than trying to select multiple files at once. I'm actually coming to multiple pairs, combos, so a list of paired. I have only one option here, and now I'm selecting that single option. So in the particular case of the tutorial, you have 40 files. In my case, I had 8. Imagine any number between 2 and 1 million, and that's the number of files you could have. This makes operating Galaxy very, very efficient, because now inside this tool, I just need to execute on that one single element and not on 8 separate elements. So please, make use of collections. They're a wonderful way of streamlining your work and making it as efficient as possible. I did talk about earlier this predictive tool suggestions. So here, make contigs is often followed up by all of these different tools. And this is one way for you to interact with your data. From memory, these are all clickable and should load the tool into this middle window, which it's doing right now. So that's absolutely fine, but we're not going to go down that route just now. So this is about empowering you to do work efficiently. We're not yet up to metagenomics at all. We're talking about just good data practice in Galaxy and how to handle multiple data sets at once. So we have constructed the make contigs file. Now we could go on and do a number of pieces of work. And this is where we want to be next. So I'm actually going to switch gear a little bit, and we're going to talk about more efficient ways to do work. So you can see my file here has now made a number of output options. I can inspect any of these. They are whatever particular data set and style is produced. In this case, make contigs merges data by name and group and gives it IDs. That's absolutely fine. I'm actually going to switch back now to my pre-computed history because I just want to show you this in a little bit more anger on a larger amount of files. So we have just constructed basically a list of files associated with the collection of fecal matter from mice on different diets. And this is where our metagenomics journey starts. It starts here, and over about 2 hours it gets to here. And it's great. I give this one all the time, but it is laborious. So we need to clean the data, then we need to map it, then we need to reclean it, we need to truncate it, etc., over and over. So first of all, we need to filter by length. Then we need to remove low quality. Then we need to understand if they're duplicates. And we can do this by executing a number of sets of tools. We do the tool, we look for the result, we do it again. That's absolutely fine. The reason this is the short version of the tutorial is we're actually going to make use of workflows. And workflows is what I really wanted to emphasize in this particular session with you. So what's a workflow? A workflow is a set of tool descriptions that are chained together and are best shown by example. So I can copy this URL by right mouse click, copy link address, or download to my computer. There's no need for me to download it, just to push it back up to Europe. So I can come back to my Galaxy Europe instance, my workflow menu. Now my workflow menu is populated with previous work I have done on Galaxy Europe, so yours won't necessarily look like this. And my options are create and import. In this particular case, I'm going to import a workflow. And I'm simply either choosing a URL or choosing a file on my computer and importing this workflow. I'll just let that run. All right. So it's come in. You can see I did this today, so I was doing a bit of pre-work before this session. Let's have a look at workflows, because again, this is really sorry, I'm just reading the questions. Yeah, I think I'm looking forward to getting into those later. So you can run this workflow. I'm going to. Hello, Prash.
Yeah, get it. Yeah. So you may go ahead, please. Yeah, you may just come back to the Q&A session later. Yeah. So Anvesh, I appreciate your patience, please. Yeah. Let Gareth finish that. Yeah. Thank you so much.
Yeah, that's all right. No, I'm just getting happily distracted. Yeah, I've probably got not too much longer, because I think we've got about 30 minutes left, don't we?
Yeah, about 24 minutes. Yeah.
Perfect. Yep, we're on time then. So you can run a workflow. I would always strongly encourage you to edit a workflow. And by that, this is the first opportunity you get to see what's inside a workflow. And this is where I come all the time to have a look at a workflow. So I'm going to spend a few minutes talking about these, because we're not necessarily going to wait for them all to execute, but this is where Galaxy gets its power. So the workflow language, canvas, and on that canvas you can zoom in and zoom out, and you can also navigate around the workflow here. It's a graphical interface, so you have inputs, and an input goes, well, this is always an interesting language, the output of the input goes to something else, and you're simply specifying how to take this piece of data and pass it to another component in your workflow. So we're taking something called contigs, and we're utilizing it twice. We're sending it to a tool called summary.sequences. We're also sending it to a tool called screen.sequences that also needs a second input, which is our group, and that comes in here. The right-hand side of our screen is the tool menu associated with the tool I have clicked on. So you can completely configure this tool and utilize this workflow at those configurations over and over again by simply running the workflow. So I do really like these. For those of you who've not seen them before, I'll give a brief demonstration of how they actually work in a few minutes. But this particular workflow is taking our two inputs. It is summarizing the quality of the data. It's screening sequences based on quality. It's deduplicating those sequences based on unique. And then it's summarizing all that by counting this material at the end. So you can see you've actually starting with only two files, passing that through one tool, two tools, three tools with two separate quality control metrics. And this, to my mind, is actually a relatively simple workflow, but it's got an awful lot in it and an awful lot that you can do at once with your data. So there's no reason I can't run this workflow, which should load. I'm just waiting for it to do that. And it's going to run on the data that's available in your history. So in my particular case, this history is pre-computed, so it has a lot of files that conform to these settings. but I know that the ones I need are way, way down the bottom, which is make contigs, trimmed. Sorry, I borrowed this from a colleague. They've hidden this make contigs group. There it is. But I've selected my contigs, selected my groups. All my tools are listed under here, except now my execute button is at the top. There's run workflow. I will run it, but we're not particularly interested in the result. I just want to show you how it works. And the workflow will run. So all the steps will be queued up. You'll get a understanding of how they work. But really what I wanted to show, and I'm a bit aware of the time, so I'll speed up a little bit more, are two elements to the workflows and then to the workflow reports. So I'm going to show you what happens when I bring in a second workflow, which I just need to go down and down the tutorial, find the second workflow. I want to bring this one into Galaxy, import workflow number 2, and then show you the real power that you can get out of workflows. So I'm going to bring in this second workflow, import. Now we have our two workflows. So I could wait for this first one to finish and execute this second workflow. The first one's running in the background now, and do this second one. Let's just have a quick look at the second one, just so that we can see where we're going with this. And as it loads here, this is what the second workflow looks like. It's a little bit more grandiose than the first one. It takes 2 inputs, align sequences and count table, and does a large number of transformations on this data. Now, in this workflow, in this tutorial, there are 7 workflows. So I could iteratively go through them one at a time. I think one of the nice features about workflows, and this really comes to the heart of one element of bioinformatics, which is modular analyses. I'm going to create a new workflow. I'm going to be a little lazy and just not even name it, because I just want to get to the result. So unnamed, that's fine. It's very poor practice for me. So what I actually can do is I can bring in an entire workflow as a single executable entity, which is my workflow number 1. I can bring in workflow number 2 as another single entity. It's quite a large one because of the number of things it outputs. And then I simply need to find my outputs here, my count table, connect it there, my aligned sequences, which is unique sequences from memory. Yeah, that'll do. To in my aligned, specify my 2 inputs, because the inputs were contained in here, so this is actually a little redundant. One is contigs, duplicate that input. One is groups. Name this input groups so I can see that. Name this input contigs. And now I've just successfully created a workflow that chains one complex set of analyses to another. And there's in fact no reason why I couldn't build all 7 workflows together as a complete result. Now that would be quite ambitious of me, but it's trying to show you that you do have this opportunity to look at your analyses, maybe in a QC phase, an assembly phase, an annotation phase, and build a workflow for each of those. And then once you're happy with it, en masse, you're going to produce a large workflow. And I can show you what that large workflow looks like by extracting a workflow out of this complex history of nearly 300 results. I can tell you on my screen, it's going to look a little ridiculous, but let's have a look at it anyway. And here it comes. Let's And there it is. You can already see down the bottom of my screen by the nature of this picture that this is an extremely comprehensive workflow. All 7 workflows demonstrated as individual tools from raw data here on the left-hand side through all the processing and visualization. But that is a complete metagenomics workflow that you could utilize yourself. So I am keeping an eye on the time. I'd like to show you 2 final elements of this particular work. And since we have a number of quite interesting questions, I think we should then transition to opening for questions. So just as a bit of nicety at the end of this particular workflow, this is one of the results that is generated from the data. This is a Kroner plot based on each individual sample. So there are 19 samples in this list here. The distribution of bacterial types, and this is an interactive HTML plot that you can explore for the levels of the bacterial diversity in this sample. This is exportable, savable, and interactive. So this is actually quite a nice endpoint for this particular tutorial. It's not the only endpoint, but I wanted to show one that's quite visual and quite dynamic to give you an indication that we're not always just ending with a flat tabular file of values. So that's pretty nice there. The other thing I promised to show is workflow reports, and I made up one of these before this particular session today. It's a fairly simple workflow report. It simply lists the Galaxy version, the time this analysis was run in local time, and extracts out one heat map from the history. How you actually go about doing this is you edit the markdown language associated with this particular history. Those aren't working, so I'm just going to get rid of that particular information. Save this history. I could do job parameters as a table, and it's asking for which particular, oh, that's a bit hideous. Let's not do that one. Job metrics, I think should come up a bit better. Nope. Normally this lists the history items like this, which is what I was looking for. I just go for a text file, we've just embedded into here, save this, and visualize this particular result. And this is, I'm going to say, about as easy as it gets to generate a workflow report. I've tidied up my Galaxy version is now listed, the embedded data file type is listed, and the image is listed. Now, I would like to make this a little nicer, and this will be my closing bit so we have time for questions. So I'm just going to bring in, sorry, go for it, Prash.
Yeah, no, great. Yeah, thank you. So are you open for Q&A now?
Yeah, I think so. I was simply just going to upload a file and show what a report looks like.
Yeah, please. Yeah, please.
But, I can get to the questions as well. If I can't find this file in a few seconds, I will give up because I thought I'd put it somewhere on my computer. There it is. So I've just specified a local file to import into my history. This happened pretty quickly and we can get to the questions. It's going to come up here. You'll see that this local file is your conference.jpg. Hopefully it comes up fairly soon. If not, I'm going to go to questions. There it is there. Sorry, it's just taking a moment. Well, I'll start reading the questions from top to bottom, and once this is complete, I was simply going to add in, there it is, a logo to our workflow report. So here is a particular image. There's no reason I can't now come to my Galaxy page, edit the content,
Can you please mute the volume? It's going.
Come to the very top of my screen, add an image from my history, which would be number 280, save, visualize, and now my workflow report has a nice graphic associated with it. So this is one way, again, if you want to share data, that you can construct for yourself quite a complex report. You can actually put a huge amount of text in here. So anyway, with that, I think I will end some of the demonstration of data's, workflows, workflow reports. I hope I've given you a bit of insight into some of the more complex things you can do in your Galaxy instance. And I will turn my attention to the questions on the screen.
Yeah, excellent. Yeah, thank you. Thank you so much, Gareth. Yeah. So do you want me to read those questions for you, or?
Well, I have them here, but yes, some of them, I believe you feel that we may have tackled the fast queuation question.
Yeah. I think there was a fast queue question by Abhishek. I think, that was already answered by you and probably me. So we can quickly go to Anvesh Pandey's question. So he asks, suppose I wish to incorporate molecular dynamics via GROMACS, which is an open source program into Galaxy Portal. How do I do that?
Yes, so the questions, I don't mind these at all. That's absolutely fine. So yeah, I will show this by example, I think is the best way to answer this. So I'm stumbling on my words, apologies for this. And also lost where you are on my screen. There we are. I think I've got half of you. I've totally lost. Oh, sorry. Apologies. Too many open screens. Okay, what I was trying to say is that traditionally Galaxy has developed tools. You've had a sense of, when I've gone through this, how a tool is laid out. It's a particular set of metrics with or without many configuration options, and you're done. You just move on from there and run that tool. And the questions we've been getting today are much more about what we term interactive environments. So places that you can explore data or look at it, or maybe are more demanding on it. They're not more challenging for Galaxy. In fact, there's something that the Galaxy community is getting into. So I want to show it in two different ways. So one is interactive tools. So these are pieces of software that generally don't fit into this very nice middle window. So they're quite complex and interactive. They may have graphs with sliding bars on them, 3D rotations, huge amount of configuration options, and they're difficult to represent in this particular format here. So now an interactive tool is an environment that you enable Galaxy, and of course I can't do it because I don't have that particular file type there, so I'll do something generic like RStudio. And what's happening in the background is a small virtual machine is being built for me with a temporary IP address, and it is being made available to me and to me only to do my work in. I need to find these through interactive tools. Here it is here. It still hasn't fully spun up, which is not a surprise, but nonetheless, it's there and it's ready for me to use. No, I don't want it stopped. Yeah. Anyway, that's not working at this point in time. I won't go too far down that route, but the interactive tools may be a place to explore those molecular dynamics. And by extension to that, we are trying to bring on a tool into Galaxy at the moment called AlphaFold. And AlphaFold is a visualization tool for 3D protein structure, and it will be inside one of these interactive environments, is what we understand, because that is the most reasonable place to present it to a user. Now, the other half of that user's question is, can we submit jobs to it? I think that's what it's asking. The answer is yes. So if something can be made into a tool or into an interactive environment, then you can absolutely use Galaxy to, yeah, so these are all just taking a minute to spin up. I won't waste time doing this. Use Galaxy to do as a batch job submission tool. Sorry, apologies. I was hoping the interactive environments would work a bit more stable, but they're not. They do take a bit of time to spin up. Okay, I think we have some more questions. Are there any tools available to predict small interfering RNAs and microRNAs? Yes, there are. Do I know exactly which of those tools they are? That's a little bit harder for me to point to right now. They will either be under your RNA-seq analysis. Let me just clear that off. RNA analysis. There's a large number of tools under RNA analysis. Again, sound like one topic person. I would encourage you to go to the Galaxy training tutorial. This is always the best place to look for quite clear descriptions. See if there is anything under transcriptomics on small interfering RNAs, which I'm not entirely sure if there is. If there were, differential abundance testing for small RNAs, here's the hands-on. Go through and have a look at what tools are utilized in this particular tutorial, or just run through the tutorial. But that would be the best place to find that information first off. Two other questions, and I'm quite fascinated, and thank you for this opportunity, because the driver of the Galaxy India community seems to be a lot about molecular visualization and molecular dynamics. I think this is quite fascinating because we haven't seen that so much in other Galaxy servers. Yes. Is it possible to create a Galaxy program containing all the available molecular visualization tools into a single platform? Possibly, but maybe not the best use of Galaxy. I would say rather the way Galaxy works, you would have each visualization tool as a tool, and then what you would do as a user is create a workflow to put your single data through all of those tools at once, and then you have all the results, because not necessarily is everyone going to want to run every one every time. So then the better way to manage it is as a user through workflow management. And the final question, I think we have a few minutes left, is another one that I'm going to fall back and say, please use the Galaxy training tutorial. The question is about rule-based import, and is there a precise way to define the rules if we're importing rule-based data? Yes, there is. So this is this particular section up here of Galaxy. There is actually, because I've done it myself recently, quite an awesome tutorial on, oh, I've got to find where it is now, on rule-based imports. And simply, the answer is, go and do the GTN tutorial, and you will be an expert in rule-based imports by the end of this. Here we go. Rule-based uploader, the advanced one is quite good. Either of these will be fine examples of what you're trying to achieve. So yeah, that's all contained there, and it's actually a really nice tutorial. I followed this one myself not that long ago.
This is absolutely a perfect setting post lunch, Gareth. Thank you so much, for a wonderful talk. It's always nice hearing you. So, friends,
thank you.
Yeah, friends, you'll agree with me that Gareth has been a fantastic speaker. You can even watch his videos in the Galaxy Australia page. he has really taken an overview of the entire front of Galaxy, right from its scalability to an overview of various tools. And I reckon I also agree with Gareth that many India users are in fact interested in chemoinformatics. That's because from our survey, over 80% of the researchers are chemists turned bioinformaticists. Right from our so-called bioinformatics practitioners. Right from 1980s or 1990s. So one important thing that you could really learn from Galaxy, largely from Galaxy Australia, is you need to inculcate a kind of a habit to interact with development team. So that plays a very, very important role. It's not that you use these particular workflows for yourself, but if there are any bugs, if there are any things that you would really like to incorporate, or if you really want to come up with your own workflow, you yourself can be a kind of a vivid developer, and you could as well interact with the Galaxy team, and that's where people like Gareth are meant for. They're absolutely great scientists and teachers. They help us move forward in reaching the pinnacle of success. Especially the most fascinating time that I've had today after listening to Gareth is he has shown Galaxy India sub-instance of Galaxy Europe that made us feel that we have been a part of India. That's absolutely fantastic, Gareth. Thank you so much. So friends, I would probably request everyone to unmute your microphone for quick 2 or 3 seconds and give a thumping round of applause to Gareth for his talks today, so that he could hear it, and also his wonderful cat could also hear it. Yeah. Thank you so much.
Thank you.
So thank you so much, Gareth. Would you mind taking a quick? Is it okay if everyone can switch on their video so that we can have a quick photograph? Yeah. I hope Subbu and Uday can help me take a good photograph. Yeah, please. Yeah. So all of you can quickly switch on your camera, please. Yeah. Yeah, we also have Chris. Yeah, thank you, Chris. Yeah, for joining. Yeah, yeah, thank you, Sangeeta. Yeah. Yes, everyone, please. Yeah. So, Gareth, is it too much to ask you to show your cat?
Oh, I think she's just had her dinner.
Okay. Yeah.
No, apologies. She has dined and fallen asleep somewhere.
Oh, it's okay. It's okay. Yeah. Please say my hello to him or her. Yeah, I'm sorry.
We will. I will.
Yeah, please. Yeah. Thank you. So, I think everyone has got shy genes here. No one is opening up their videos here. But let's assume that they have their photographs here. So please, yeah, say cheese. Yes. 3, 2, 1. Yeah. Thank you. Thank you so much, Gareth. Yeah. So friends, moving further, we're absolutely on time. It's 3:01 here, India. So we have Christopher Barnett, who is going to be the next speaker. But for that, Sangeeta Savan, who is the director of University of Pune Bioinformatics Center, should be chairing the session. I will request Dr. Savan to take up this particular session. Thank you so much. Yeah. Sangeeta, would you mind unmuting your microphone?
Yes.
Thank you.
Thank you, Prash, for giving me this opportunity to chair this session. I'm very happy to do this in this particular workshop. I believe this has been a wonderful workshop which has been arranged by BioCruise, and I'm really happy to be here. I have this wonderful task of introducing Dr. Christopher Barnett, who is the speaker in this particular session. It's a privilege to introduce him. Dr. Barnett is a faculty member at the University of Cape Town in South Africa, and he is a very accomplished researcher and professional with a NRF Y2 rating, and his interests in the research include basically glycobioinformatics, chemical glycobiology, data analytics, visualization, and the repeatable research with the Galaxy project. He's basically interested in the interdisciplinary research, which is at the interface of chemistry and computing, and that's very interesting. He is focusing on the molecular recognition and binding of carbohydrates and proteins. So the glycans and proteins. That's the focus area in which he is interested in. He also he's skilled in molecular simulations. He's also an ardent programmer, so he's interested in writing scripts, programs, and doing the data and project management. And as per his biodata, he is also very keen in sharing the software with others. So that's very generous and nice kind of hobby, I would say, you have, Dr. Christopher. It's really nice because sharing is very important in this era of collaborative work. So with that, I would request Professor Dr. Barnett to deliver his talk, which is titled as Structural Bioinformatics and Chemoinformatics Using Galaxy. So over to Dr. Christopher Barnett.
Sangeeta, thank you for the very kind introduction. Much appreciated. And thank you for the invitation from the INBIX team as well. I'm just going to set up sharing and hopefully give me one more second while I make sure that is functional. So let's just see. I hope everyone can see that. Maybe Sangeeta, could you let me know? Can you see that coming through?
Yes, we are able to see the screen. Yes.
Fantastic. Thank you. So let me start. So hi everyone. It's really great to be able to present to you. Sadly, it's only virtually. So maybe one day I will be able to travel and see everyone in India. That would be fantastic. And just again, thank you for this very kind invitation to share this work at this Galaxy workshop at INBIX. And what today's presentation is about is the structural bioinformatics and cheminformatics tools that are available in Galaxy. And I think that aligns with the idea that I think that most of you are interested in strengthening your drug discovery background. So there's a really broad space there, which includes running molecular dynamic simulations or doing screening, etc. And this work has been done in collaboration with Simon Bray, Tarindu, Bjorn Grunning, and I'm sure you've recognized some names here from the Galaxy Europe team. So I'm really grateful to them as well for all the hard work that they put in to make sure that these tools are available. Okay, so in today's talk, I'm basically going to give you an overview of what's out there in terms of our training, and I'll give you some examples as well. And then just in terms of if there's any questions or any problems during the talk, I'm doing the usual juggling a few screens. So I'm going to ask Sangeeta if you could help me. If there's any issues, will you just let me know, and I'll try to attend to those.
Sure, Chris. I'll do that. Thank you.
Thank you.
Yeah.
So let me move on. So that's the general background. Let's see how Galaxy can be useful to you in a drug discovery setting. Okay. So you probably already know the answer to why Galaxy, but let me take a moment to discuss it a little bit further. And I think the first useful reason, or the reason why you'd want to use Galaxy is that it's a web-based platform, so anybody can understand how to use it, and it's accessible from many devices. And in terms of research then, it just means that from your undergrad students, I mean, even from high school students, the public can be contributing to research as well as more experienced researchers. So I think that's probably a really key feature there. The other key feature is you've already seen from Gareth's talk that this is a tool that supports multidisciplinary research. So whether it is transcriptomics or whether it's cheminformatics or perhaps even related to climate modeling, this project supports all of these different disciplines. And of course, today I'll speak more to the drug discovery pipeline. And you can see a number of the other reasons to use this project. And I'm sure these will jump out at you because during your work in drug discovery, and maybe if you're starting out, you haven't seen this yet, but you'll note that with research, you're always struggling to figure out how can you optimize processes, how can you share data, how can you make things accessible. And I really think that together with the community of Galaxy, the software then helps one do such, do this kind of work and carry out useful research and share it. Okay. There we go. So you might already have an existing computational drug discovery pipeline. I'm not sure. And if you do, this might look familiar to you already. And if you're not there yet, this is a fairly common pipeline. And there's a few approaches that you could take, but generally there's a protein target that is found through multiple methods. And then the computational side of it that's interesting for chemists is, can you design structures that would then target the binding side of that protein? And you can use a number of methods to do that. So you could look at ligand docking methods. You could look at running simulations to see if maybe the protein has some interesting movements that you could, maybe you find that useful in your research somewhere. And you might then look at how the ligand binds to the protein as well. So there's a number of strategies that you could use, and you could imagine this section of the work as well as being quite repeated. There might be a sprint, you might be repeating this several times before you get onto further approaches. And you also might find that even if you get to after your lead optimization, you might find there's a problem and you might need to come back to these stages. So these are routine methods that I think would be used in the drug discovery pipeline. And Galaxy, I'll speak to you today about the ligand docking and the MD simulations more than anything else. But Galaxy does have these tools. And where it doesn't, or where you feel there should be an improvement, because we have this beautiful community of people that are really invested in making sure things work, you can liaise with someone and add a new tool. So there's a lot of opportunity for contribution and collaboration here as well. So in terms of what Galaxy currently has to offer, there are multiple subdomains on Galaxy EU, and there are in fact several public Galaxy servers. But I'm going to particularly talk about EU quite a lot because the work that I've done has been in collaboration with the Freiburg team, so the Galaxy EU team. So if you go to the Galaxy EU website, you might choose to go to the cheminformatics subdomain, and there that will show you all of the computational chemistry and cheminformatics tools that are available to you. And of course, you can access them from any other part of the website as well. But this sort of subdomain helps you just make sure that you're seeing the tools that are relevant to your current project. So there's a few papers here that might be interesting to you. And one of them is the Chemical Toolbox, so by Bjorn Grunning. And there's a few others that speak to creation of these tools and function. Okay.
Sorry to disturb you, Chris.
Yes.
Is it possible to zoom in a little bit, because there is a comment saying that they're finding it difficult to read the screen.
Oh, okay. Thanks for that comment.
Yeah.
Let me just see if I can figure out how to do that. Let me try reshare. So I'm going to stop sharing and let me just try reshare this. Let's just say, okay. Let me try something a little bit different.
Chris, you can use some option called as change layout. Then you can use Spotlight.
Ah, is that within Google Meet, or is
that
it
is in Google Meet? You can see the three dots there. More options. Go there and just click on the change layout and select Spotlight.
I see. Ah, that's perfect. Thank you. I'm going to reshare and try that out now. Give me one second. Thanks for the help there. Okay, let's do that. So, okay, I'm going to... Okay, so I'm redoing the share, and then give me 1 second while I change the layout again. Okay, change layout and spotlight. Okay, I think hopefully that's going to help. So I hope that's a bit better and clearer. I don't know if I can do much better than that at the moment. I've just happened to have a very wide second screen. So does that help everyone? Okay, fantastic. Okay, so let me continue from here. And yes, I'm also happy to provide slides. So if anyone wants those after the talk, I'm very happy to provide those. So okay, so this is the Galaxy Cheminformatics view. So if you go to the website, you will see this, and I'll probably zoom in a little bit more later as well. Right, so this is then through the work of the Galaxy EU team and also the Galaxy Computational Chemistry group, which is on GitHub. So there's a few tools there that I think will be very useful to you in the drug discovery space. Okay, so what about, if you think about chemistry data, I'm sure I've missed out one or two facets of it, but there's often atoms, connectivity, you might have Cartesian data, you might have properties of the system, and this might be in various formats. And you also might have databases of molecular properties and chemical names. So there's a lot of interesting data to deal with. So for example, if you've got a number of drugs you're looking at, it might be in this kind of form, but of course as a database, or you might have a protein and a ligand and multiple Cartesian data. So can Galaxy really handle this? And the answer is yes. And the reason the answer is yes is because of the work by Galaxy Computational Chemistry and the Freiburg team. So I'm going to repose this question, or maybe pose it for the first time, is why Galaxy? And the reason I think that's really useful in a drug discovery context is batteries are included. So there's so many features that are useful for the research journey, which include automation and sharing, being able to create histories and share them and create workflows. You have public resources, and you've also got the opportunity to run this internally. So if you have a computer in-house and you want to test out new ideas, you can also do that. So there's so many ways that the software behind Galaxy allows, fits, checks a number of boxes for drug discovery and that pipeline, the drug discovery pipeline. The other thing I think I've sort of mentioned is data typing. So the formats, the file formats that we work with in drug discovery, Galaxy helps us handle those better. So what I'm going to do over the next few slides is just talk again, or talk a little bit more about some of these features of Galaxy that I think are really useful in drug discovery. And then I'll go over very briefly some theory of docking and molecular dynamics and some examples. And I'll end with a large example, which I think once again showcases how Galaxy is useful in this space. So imagine you're busy with your drug discovery pipeline. You might have many molecules that you'd like to test. So you can imagine many calculations. And how do you track this? So this is something that Galaxy can help you do. And this might be a bit intimidating to see, but this is a picture of the Galaxy website and the history view and a number of histories. So I'm just going to click through them. And there's a whole lot of histories at the moment. Some of them, the jobs have completed, they're in green, some are running, some are in gray. And what's fantastic about this is that these might have been done through an automated process, and in fact they were. This is courtesy of Simon Bray. And what you can see here is that a researcher is able to run multiple calculations, or maybe you could rephrase that as multiple research hypotheses in parallel, and get Galaxy to track and manage those. So it means there's a reduction of the effort trying to manage this yourself. So I think that's a key feature of Galaxy. And of course, you can share these at any time. Another key feature is you can see that some of these look quite similar. The process for all of these simulations is probably the same. It's probably the same workflow. And what that means is once you've tailor-made a particular process in drug discovery, so you've decided you want to use a bunch of tools in a certain order through working in your history, you might then have created a curated workflow that solves a particular problem in your drug discovery process. So once you've got that up and running, you can share that workflow, and you can also then repeat the same process. So why that's useful is if you want to take 100 or more ligands and run molecular dynamics, instead of doing that by hand every time, you can figure out the process. And once you figure out the process, you can just repeat that across your entire set of data. So you can streamline your data creation and your data analysis. And the other side of it, of course, is sharing with other people, and reproducibility and automation, which I'll touch on a bit later. And then the other thing, I think, as a computational chemist, so my background is as a computational chemist, and sitting in front of a workstation in Linux, trying out all sorts of ideas. It's really important to have a space in which you can not only automate, but also visualize what's happening. So I still do use tools outside of Galaxy at the moment, but I think Galaxy is moving towards being a great space for visualization, where you can look at many formats. And this is not only for plotting graphs and data, but also looking at your structures. And then more recently, Galaxy's included Jupyter Lab. So what that means is you have this opportunity to interactively plot and access your data set. So I think that then meets this need of for any scientist, but particularly if you're in drug discovery, to try out new ideas and iterate on them and figure out where things are going wrong, and have a playing space as well, where you can try out anything, but then you can also go back and create these workflows and automate the processes more quickly. So just to sort of finalize this section before I go into some of the methods, is if you're interested, I'm sure you already know this link, but training and upskilling, there's the training.galaxy link that you can go to. And in a moment, I'll show you some of the trainings for computational chemistry. And of course, this is all open science, so you can find all of it on Cheminformatics and also through our GitHub. And I can share that maybe later if you don't have access to this link, or if you can't see it so well, I can share that later as well. So if you want to contribute code, you're most welcome to go to our Galaxy computational chemistry repository. Okay, so this platform allows sharing, it allows curation, it allows all these wonderful things. And I think a lot of people, when they first hear about Galaxy Project, they wonder, how can there be so many wonderful resources available? And so I suppose the answer is there is some good research funding happening in the background here. But what's really fantastic is there's this public resource that we can all tap into. So again, I'm speaking to Galaxy EU. Of course, there's other public galaxies, and Gareth probably mentioned Galaxy AU in his talk. And what that means is, behind the scenes, as a researcher, you don't have to worry about the compute resources. You just submit the calculation that you're interested in running, and what Galaxy will do for you is do the scheduling and do the queuing to all of these high-performance computing resources. If your calculation needs to use different types of compute, so not just CPUs, but maybe some GPUs, Galaxy is able to schedule this appropriately. And where it doesn't, you then, of course, reach out to the community and say, hi, I'm trying to solve this kind of a problem. I have a very large data set and more GPUs. Can you make that possible? So I think behind the scenes, we're very lucky to have all of this public infrastructure. Okay, so that's just a brief overview of what's happening for Galaxy EU. Now, in terms of what's inside this chemical toolbox, so this is a great paper by Bjorn. He started looking at the purchasable drug space, but also creating tools that are on Galaxy for computational drug discovery. So what we need in that toolbox are a number of tools. And in this fairly recent paper, you can see what that toolkit looks like. So what you need to be able to do is upload data using these upload tools. So we've got opportunities to upload SMILES data, Cartesian data, etc. You can pre-process that information using a number of available tools. And this is not all of the tools, of course, they wouldn't fit on one slide, but just as a hint of what's out there, you can do some pre-processing. You can also start exploring your data. So, drug likeness, NP likeness, all of these molecular descriptors. Then I'm sure many of you are very interested in simulation. So looking at doing some docking or doing some molecular dynamics, you can do that with Galaxy as well. So all of the simulation tools to set up simulations, to run the simulation, and then to do the analysis are available. And then I've briefly mentioned already the visualization, so that's also there. So this all links together to be, I think, a very valuable resource for conducting drug discovery and general chemistry research. So looking at that platform, I hope many of you have already done the Galaxy 101 training, so this will look highly familiar to you. On the left, you've got all the tools, and if you need to find tools that are not readily apparent, you can use the search toolbox over here, or the search bar. In the middle, you've got access to all the tool parameters. And for some of the MD tools, this will look a little bit complex, but through the trainings, you will see how easy it is to run these many simulations, and that the parameters are actually not too bad. So there's opportunities to select the relevant data sets, select relevant parameters, and then of course run the calculation. And you can see on the right-hand side, here's an example of a history, and this is for a particular project where it's the T4 lysozyme. And in that project, the ligand is benzene, and the protein is this PDB ID, I think it's 181L, that's been fixed already. And from there, this whole process goes forth, and you can see these jobs are still running. So of course, you could switch to another history in the meantime. You could do all sorts of exciting things. But that's the general space that you would be working in. And then of course, this is not all of it. You've also got at the top your workflows, your shared data sets, and a number of other features. So there's so much more to dig into and learn about with Galaxy, but this is the basic framework to get started. And for many users who have not done coding before, this is really a valuable interface, because it is really tricky working on the command line if you're new to a discipline or if you don't have experience with it. So I think this allows many users to get up and going with the research, and also make the right kinds of mistakes, not silly errors that we don't want to make, the right kinds of errors, and to learn from those errors. So talking about learning, if you're interested in learning about particular parts of computational chemistry, we do have some training already live on the training site. So we've got a few tutorials, and just after this talk, Simon Bray will be giving a tutorial, and under correction, let me just see if I've got the name. I don't think I have the name on me, but under correction, I think it's going to be the high throughput tutorial. Let me just maybe double check that. Sorry, excuse me, it's the protein ligand docking tutorial that Simon will be taking you through later. So if you're excited to learn about that, I encourage you to join him after this talk. But of course, all of these trainings are available, so you can at your own pace access them and try them out. And if you're having any trouble, you can chat to us on Gitter and ask for support. Okay, so I've done a fairly brief overview of why Galaxy is useful in the space and what material is currently available. And maybe I haven't mentioned one or two things, so I encourage you to ask questions to see if there's a particular tool that you're wanting to use, if it's there or not. Or if you're wanting to develop new tools, we can speak about that in the chat as well. But let me briefly talk about some of the methods that are available. So I'm going to talk to you about docking, molecular dynamics, and free energy. And those are three methods that are commonly used in drug discovery. And it will depend on your pipeline. So a lot of labs will pretty much only use docking. But if you have a little bit more time, and this does depend on your project, you probably would want to run some molecular dynamics as well. And if you have a bit more time than that, and you really want to use a better method, you'd maybe go towards free energy. So I'll speak to each of those. So docking, I hope, is very familiar to all of you. It's the simplest way of thinking about it is as a lock and a key. So you can imagine that the enzyme is the lock, or the binding site of the enzyme is the lock, and we're looking for molecules, we might call them ligands, that are keys, that in other words fit the lock well. And in fact, instead of opening the enzyme, we're often looking to stop the enzyme from doing something. Okay, so we put the key in and we want to stop the enzyme from doing something. So this strategy is really, really useful for post generation and for quick screening. And you can complete many thousands of poses, and many thousands of molecules can be docked in a very short amount of time. And depending on your docking process, you might have multiple levels to your screening, so a high throughput part, and then you might sort of funnel and filter down. But you could probably very easily, if you know the system quite well, do this in a few hours or a day to get your initial results. And then I think the tricky part is then deciding if they're valid to take to the next stage. But yeah, the actual post generation and screening can be quite quick, depending on the system and if you've got good force fields, and depending on the size of your database. But yeah, this is what most people want to do. And there are tools within Galaxy to help with docking. And as I mentioned a few moments ago, we have a protein ligand docking tutorial. And in that tutorial, which Simon will go through later today, you'll learn about cheminformatics in a bit more detail, at least this facet of it, and particularly protein ligand docking. And then you'll also learn how to do that in Galaxy. So you'll perform a simple docking workflow in Galaxy. So you'll work with a bunch of molecules. Here's some examples here, and you'll dock them into this heat shock protein, which is I've shown in the background here, and here's some of the molecules that are docking into it. So you could think of that as a very important first pass in your drug discovery pipeline. So if you're interested in that, I encourage you to join later. Okay, so what's beyond docking? Beyond your initial docking, you might choose multiple levels. You might go for using different parameters and cycle through this docking process. You might use different tools, and you also might choose to score your molecules in different ways. But in fact, a lot of people would want to use these initial poses and then try figure out how the particular ligand moves over time in the protein. And you might ask the question, well, um, does the conformation of the protein change when the ligand is there? Does the ligand unbind? You can ask many, many complex questions. So I think the first sort of basic idea, though, would be let's look at this reality, which we maybe can't probe experimentally all the time. Even from crystal structure, we're only getting static screenshots of a system. So let me just run that again. We want to effectively, in some way, create a molecular movie and see what's happening over time. We want to get resolution over time and space in the binding site and sample the positions of atoms and examine things like conformational change, structural and temporal properties. And there's so many things you can do in this space, but for a standard, basic run-of-the-mill MD simulation, we have some tutorials for that. And in fact, we've also got this high throughput MD tutorial. And what you'll do in that is take a protein and take a ligand and perhaps multiple ligands, and then build up multiple boxes of water with the protein and the ligand and run that over a period of time. And of course, in the training, it'll be a short time because it is for training purposes. But in real life, for a proper project, what you choose to do is run that for quite an extended period of time. And what you then might find out is the principal modes of the protein and perhaps see if they've changed depending on the ligand. You might find if a ligand unbinds, and that can be quite useful to find out. So if a ligand that scored quite well in your previous stage or docking stage unbinds in your MDE, it might be an indicator that it's not a very good inhibitor of the enzyme. So that's one way to think of that. And of course, there's many other things that you could do in this space, and you could try out some more advanced methods. But in this training, you'll do high throughput of a standard MD, which is basically then the water with the protein and the ligand in this water box. Okay, so what does that look like? I'm just going to show you. This is just a quick movie of the histories that are available on Galaxy EU. So if you want to look at these, if you don't have time to run the calculations yourself, you can have a look at the histories, you can import them. And once you've imported them, you can see what's been done along the process. I think this example here is this heat shock protein with the ligand, and then a number of dynamic steps have been run. So there's the imported history on the right-hand side over here. And those MD steps have been run, and multiple analyses have been conducted according to a workflow. So here's the initial protein, That's the PDB format, which is a text format. And of course, you can view that directly in Galaxy. So you can use some of the viewers. You would have clicked on that little icon over there, and you can see just generally what this protein looks like. And again, you could use some more sophisticated methods, but just generally, the water's been removed. So you can see the protein in a cartoon format, and you can see the ligand in the binding site. So that can be used as like quite a quick way to check if everything looks okay. And then you can conduct some principal component analysis and see just generally the movement of the protein and what the principal modes are. And then you might also want to convert between different formats. So you can do that directly on Galaxy. There's no need to download data and convert it on your own machine and then re-upload it. You can do it directly in Galaxy. And the reason you might want to do that, I think we've solved some of these problems more recently, but some of the tools might not support all formats. So you can convert between them and then perhaps run some hydrogen bonding analysis. So you can take the existing histories we've got and you can try out some new methods on them if you like, or some new analyses. And then I'm just showing here quickly, imagine you had a history, you've done a lot of hard work and you want to create a workflow. It's really simple. You click one button and a workflow is created for you. So that means you have the freedom to try out a lot of ideas in a history until you're happy, and then you can export whatever level of complexity that workflow is. You can export that and then share that with your colleagues. And of course, rerun that yourself. So there's many great things that you can do directly within Galaxy with these workflows. So if you have a look at the workflow panel at the top there, on all of the Galaxies, I believe there's some new exciting projects going on to make workflows more universally accessible. But for the moment, on each instance, there will be some published workflows. And for example, you might see published workflows and you could use those. And of course, you can also publish your own workflows. So if you find something really fantastic and you want to share it on a Galaxy, you can just make your workflow public and just share that with everyone. So another great way to share your research with your fellows is through these workflows. Okay, so those are the two points that I want to start with. So the docking and the MD. And I just want to take a pause. I don't know if there's any questions at this point. I'm just going to go back to the Google Meet window and just see if there's any questions from anyone at the moment. And then maybe I'll take a few questions and then just move on with the next part.
Shall I read the questions for you, Chris?
That would be fantastic, please, Sangeeta.
Yeah. So there is a question here. Shivam Pandit asks, can we run around like simulations around 40 nanoseconds or more in Galaxy?
Right. So Shivam, thanks for that question. The short answer is yes. The longer answer is you might find if you have any problems with that, let's say you have a problem with that for some reason, you would just contact the team and they'd make sure that your account has all the required support to make that run. But the short answer is yes, you can run these longer simulations. And what it's quite common to run, let's say, a shorter simulation and then replay it. In other words, rerun the tool again and extend the simulation. So you might have a workflow that runs a 10 nanosecond simulation followed by another 10 nanosecond, etc. That's probably behavior that I've seen quite a lot, is that people tend to run these smaller chunks and they chain together. So yeah, that's what you can do that definitely.
That's fantastic. So next question from Divya Jindal is how binding energy is different from free energy.
Oh, great. Good question. So I think in this context, I'm probably referring directly to the free energy of binding. And I think you can slice that up and you can do some exciting things there. I mean, you can think about the free energy of larger processes, but often in drug discovery, you're probably most interested with the free energy for the binding process specifically. And while that can get kind of complicated, we're going to look at it specifically for a ligand binding to an enzyme, and I'll speak to this in a second, relative binding free energy. So the difference between one ligand binding versus another. But yeah, that can get quite complex. So I think I'm just going to say it's the free energy for the binding process is what we're looking at as much as possible. Yeah.
Shall we go for one more?
I think so. Yeah.
Okay, so it's the next one is from Binta Varghese. She's asking, can we use polymers as ligands for docking?
Okay, great question. Yes, you can. I definitely think that you run into a few issues. So this does depend on the docking software itself. And definitely, I've once or twice had some issues with peptides as ligands. So the short answer again is yes, but you might, as with any system, hit some system challenges, and that might be to do with your force fields and things like that. But yes, the answer is you can use polymers for docking. Yeah.
Yeah, so there is another related question which is from Divya again. So it says, can we perform peptide ligand docking on Galaxy platform? So I think you have already answered that.
Yeah,
But if
maybe
you
I
would
can
like to
just
comment further, yeah.
I can extend on that a little bit,
Yeah,
Sangeeta,
yeah,
and
yeah.
just say that I've run some peptide protein simulation. So my ligand was a peptide, and I had a whole protein. So these are antibody and antigen type simulations, molecular dynamic simulations.
Sure.
But for the docking point of view, I've one of my students has done some of that docking, and I'm not sure if they ended up completing that on the Galaxy platform. I hope they did. But yeah, again, it's the same, a very similar answer to before. Is you might have some system specific challenges, and that's often because a lot of the software is built to understand that the ligand is a small drug molecule, and that the receptor is a protein. So you might hit some minor challenges, but I think you should be fine. There's one piece of software called LightDock that we were really hoping to get into Galaxy, and I haven't got there yet. So if that's something you're interested in, perhaps we can talk further about opportunities and all that on that side. Yeah.
Okay, there is one more. Would you like to take that?
Okay. Yeah.
Yeah.
Let's see.
This one says, can Nagamani, it says, can we deploy our own machine learning models in Jupyter Lab in Galaxy?
I think the answer is yes to that as well. So I haven't specifically used Jupyter Lab for that purpose, but I know that the Jupyter Lab in Galaxy, you can use it in a very similar way to Google Colab. So at the top, you can install the libraries you want to use, and you can set up, your Jupyter Notebook in any way that you like. However, I will say that I think there are some tools already in Galaxy to help you run machine learning models. So I'll see if I can find the link for that. But there's a training on the Galaxy Project training website for machine learning models. So maybe that would be helpful to you as well. And I hope I've been able to answer that. Yeah.
Great. So thanks a lot, Chris. And right now, that's all we have.
Okay. Great. Yeah. So let's move on from there. Thanks, Sangeeta, and thanks everyone for the questions. Let me just get back to my other screen. Okay, there we go. So the next thing, I suppose, related to one of these questions is binding free energies. So let me just get to the right slide. There we go. So binding free energies, and the reason that you might want to use this kind of a method is what is often done with docking is you find these poses of a particular molecule or set of molecules that fit within the binding site of a protein. And then the question is, how do you score those poses? Because ideally you want to score them or rank them in some way that relates to an experimental free energy of binding. So in other words, what's actually seen experimentally, like maybe one molecule binds better than the other. And you often then unfortunately find that the scoring functions that are used in docking do not always work very well. Sometimes they work great, and sometimes they don't work well for the particular system you're dealing with. And you might then choose a different scoring function, or you might try something, maybe many of you have heard of MMGBSA or MMPBSA. So it's almost a free energy method. All of these predictions to try get a number that is sort of meaningful and relates to experiment, so that we know that the order of the predictions at least is correct. So in other words, your top scoring drugs or your top scoring ligands are really top scoring. And that's hopefully what happens, right? It doesn't always happen. So when you hit this challenge of it not happening, what you could choose to do is use a different scoring. What you might choose to do, though, is look for a better method. And the better method then is to use, to try calculate these binding free energies properly. So by properly, I mean you want to calculate the free energy difference as follows. And what you need then is the partition function of your protein and your ligand system, the partition function of your protein, and the partition function of your ligand. And you can generate these partition functions, of course, by running either Monte Carlo or molecular dynamic simulations. And my preference is MD, so I'll speak to that. And so you're going to run these long MD simulations. But of course, the challenges that you will have will be related to the size of your system, perhaps the force field that you're using. But more often than not, it's that the simulations you're running are equilibrium simulations. And to really see all of the partition function, you would have to run the simulations for an exceptionally long time. So I'm just going to throw out a number, let's say 50 seconds. But when I say 50 seconds, I mean 50 seconds of like real world time that we want to simulate. So that might take a very, very long time for your system. That could take weeks or months or longer, depending on your system. And you'd also have to analyze all this complex data. And you couldn't be sure that you've actually visited and sampled the entire partition function. So it's slightly difficult. So there are a few methods that you can use to actually solve this. There's 4 possible routes, and there's a number of references in this field. So there's a few down there if you're interested. You could use a method based on histograms. So you could calculate the probability of finding the system at a particular state and try and move it out of that state using a biasing probability. Excuse me. You could also calculate forces along a reaction coordinate. So if you've got, it's difficult to define these reaction coordinates sometimes, but let's say you've got a very specific thing that you're trying to do, like break a bond or form a bond or look at a torsional angle a phase space. You could maybe then imagine ways to force that system from low energy regions to high energy regions. In other words, to sample the rest of the phase space. But that's also has its quirks. What I'm going to speak about today in not too much detail, but a little bit of detail, is using perturbation theory, which is really, I think I really like this idea because it links back to the idea of chemical alchemy. So back in the day, before we were all called chemists, we might have been alchemists or magicians or something different. We weren't scientists yet, and this field was in its infancy. But this idea of turning lead into gold, or something more valuable. So what we can do with this method is we have a perturbation method where we can change one chemical system into another chemical system. And so in terms of drug discovery, what we can do is change one ligand into another ligand. And in terms of your, if you've got a series of molecules, they could be very varied, or they could be very similar. So you might have a methyl group at a particular position, and you might want to transform that into a halogen or some other function, and see how that would really impact the binding of this drug. So this is a useful method for that purpose, and it links back to this idea of changing lead into gold. We can effectively do this using simulations. And then the final method is you can use equilibrium work systems, but yeah, that takes quite a lot of time. So let's focus on this idea. So you could use a perturbation to actually change your ligands. You could also mutate your protein if you wanted to as well, but I'll speak to the ligand example. So what you'll do then, there's a few ways you could go about it. I'm going to speak to relative binding free energies or relative free energies of binding. And the question then that's directly related to drug discovery and your pipeline is, does ligand 1 bind more strongly than ligand 2? And you can take this idea and apply this to then multiple ligands. So you can have a series of ligands, and you would want to then ask this question for the series of ligands. And what's required then is a few simulations, and the simulations will be of the enzyme all by itself in a water box. So this sort of pink, and I guess it's light purple, is meant to represent a water box with the enzyme in it. Then you can also do a simulation, an MD simulation of a ligand, your ligand 1, which in this case is quite spherical, and it's also in a water box. And then you want to also figure out, you want to get this delta A, but actually it's quite difficult to calculate. So we will actually go through this process in a roundabout way. We're going to look at the binding of ligand 1 to the enzyme in a system, which is this one over here. And we'll also do that for the second ligand. So ligand 2, which has got some interesting new feature. Maybe it's that halogen or methyl group I mentioned, maybe something more complex. And again, you run that simulation, and you run a simulation of that ligand 2 bound to the enzyme in this water box. So actually going from the enzyme and the ligand to the bound system is quite difficult. So getting delta A is actually quite difficult for a number of reasons, but mainly the reason is the partition functions of these systems won't overlap very well. So we won't hit convergence if we try and do this kind of a thing. So what we tend to do is actually calculate this free energy change. Okay, so going from ligand 1 to ligand 2, not bound, and going from ligand 1 to ligand 2 in the bound state. And it turns out then we can get these relative binding free energies. So delta A, so the change in free energy as follows. So there's the equation. And that's really useful because you can imagine once you've calculated this system, you can calculate it for ligand 2, ligand 3, 4, 5, 6, 7, and you should then have a series of connected relative binding free energies. And you don't have to add 2 simulations each time, you just have to add 1 set of simulations. So that can be really valuable and cheapens the cost if you don't have to rerun all of the simulations for each pair. You can solve this more elegantly, but of course you could run various combinations of this just to check that it's working. But this is the basic idea: is that you converge on this answer, and this should be theoretically a better answer than the other scoring functions. The challenge in this space is the time that can be taken to run these simulations. But yeah, these are very valuable, and I'll show you an example in the next few slides. So I suppose, first of all, does it work? You could ask that question. And there's so many papers in this field. Of course, I'll refer to one that my name is on. But there's so much work done in this field that's not related to this paper at all. But basically, the idea is then, can you match your free energies that you've predicted experimentally to your calculation? And the answer is generally yes. You can see the performance is not too bad. Okay. In terms of the order of the ranking order, so the order of scoring, it usually performs quite well. But also in terms of the actual error, it generally performs quite well. And this can depend a little bit on your system. It can also depend on the force field that you use. The force field you use. But generally, these do perform quite well. And there's so much in this space. If you're interested in this, there's so much literature that you could refer to other than the one paper that I've referred to here. But this really does work quite nicely. The challenge is, do you have the time for it? So this is a particular stage of your drug discovery process. You need to decide, do you have the time to do these kinds of things? And perhaps, and where it's often can be used, is if you want to validate your docking scoring in some way and say, for a certain class of compounds, the scoring from docking is looking really good because it agrees with these relative binding free energies. That might be something you could use. That's a hypothesis that you could test. So that might be one way that you use that. So hopefully, with the advent of more compute resources and more GPUs and public resources like Galaxy EU, we could be doing more of this kind of computing, because you can see from the previous slide, these systems, these calculations actually are done independently. It's only at the end where you calculate these free energies. So you can imagine pushing these calculations across multiple compute resources, which I think fits quite nicely with what Galaxy can do. So yes, of course, you can then do lead discovery with free energies. I still think this is quite fairly new in the sense that not too many people are doing it just yet. I mentioned the computational burden, but it does work really well for a number of well-known systems, so previously well-researched systems. It works really well for, and we've also shown it to work quite well for a cancer project. There's a breast cancer project that we've worked on, and I'll speak to that in a second. But effectively, you can use Galaxy, you can use workflows to set up, run, and then analyze these systems. And behind the scenes, I can assure you that there's a lot going on. So although it just looks like one simple calculation here. There's definitely a lot going on behind the scenes, but from that we can run all of these different simulations, and we can then figure out if everything converged, and if the final answer we get is something that's publishable. So, for example, let's look at a protein called sialyl-transferase. Again, I could have maybe included some more references, but if you look at this paper, you'll see some more detail about it. This is quite an interesting challenge because the sialyl-transferase has been identified as a key enzyme in breast cancer and some other cancers as well. So it's a key enzyme to target. So in terms of targeting that enzyme, though, what we see is one of the ligands that it has is a sialic acid. So the sugar over here, sugar derivative, and then there's this phosphate, and you've got a base, etc. on this side. So you've got the sugar and the base on that side. And in the particular project, what we're looking at is what happens if we change the R group. So if we change it from the carboxylate and you add an amine functionality, or if you make it an alcohol, what happens along the way, and can this be predicted and linked to some experimental work? So you can read some more detail at the link, but in short, we use a number of binding free energy approaches, and we generally found that they worked quite nicely. In some cases, the RBFE, which I've spoken about today, has some challenges if you're changing from a charge to a neutral R group, but that can be overcome if you're careful. But just in general, this performs really well, and we get the change in free energies and an error as well, so we have a feeling of whether this is good or not, and it converges quite nicely, even for quite complex ligands. So I'm not sure, maybe in the chat you might want to share the kind of projects that you're working on, but I would say that just in general, a lot of drugs would be I suppose we're moving towards natural compounds nowadays, but a lot of classical drugs are very simple and flat. Nowadays that we're using nature to sort of as an inspiration behind our drug design, I think we're sort of moving towards drugs that look more complicated and have a natural product look. So I think this is in that field, because we are looking at the ligand, which is got sugar functionality and it's got base and all of that inside there. So this is, I think, a fairly difficult system, and I think what we saw is that this binding free energy method works quite nicely even for these more complicated systems. So yeah, I think lots of great opportunities for using tools like this in drug discovery. Okay, so yeah, so just beyond the free energies, what you could also do for your lead optimization, perhaps classically you've thought of lead optimization and thought of using your results from docking and looking at the binding sites and seeing which parts of the ligand engage with particular parts. So there's these non-bonding interactions with parts of the protein. You could also think of using your dynamic or the results from dynamic simulations in this lead optimization. So you might choose to look at your structure files and your trajectories, so your molecular movies effectively, and then analyze those for patterns across time. So you might find that a particular hydrogen bond, you thought it was very interesting from your docking, but you might find it breaks very easily during this dynamic simulation. And that might help you then understand whether or not to drive your lead optimization to keep that hydrogen bond or not. So there's a few interesting outcomes there. Also PCA, you might then find from a PCA, a principal component analysis, that there's particular motions in the protein, and you need to perhaps liaise with the computational chemist and do some further simulations to understand the actual binding, or sorry, the modes of motion of the protein before continuing your binding investigation. So yeah, there's a lot you could do in this space. So I think we've got really a great set of tools that you can use in your drug discovery pipeline. Okay, so I think the last thing that I'd like to discuss today is just to showcase, within the context of the COVID-19 pandemic, how Galaxy can be used as a global platform to solve, really problems of huge interest to the public that affect us right now. And I think, sometimes the problems we're trying to solve don't have an immediate result. Maybe you can't affect someone today. And I think this COVID project is really exciting. I wasn't personally involved in it, but I'm going to talk about it. So Simon, who was involved in this, will give the workshop a bit later. But this is a great example of how open source tools and a public cyber infrastructure like Galaxy can be used to do transparent and reproducible research. So this project had many parts to it. So the genomics part, the proteomics part, and all of this is available online. It's on the following website. There's a number of videos that you can watch as well. You can also look at it on GitHub. But I'll speak to just very briefly to the docking simulations of the SARS-CoV-2 main protease. And the point there was then to, of course, dock a number of fragment hits that were actually found experimentally. So this is actually quite an exciting project. They were found to experimentally bind to the protein. So from that experimental data, those fragment hits, can you use a computational approach, a docking approach in the drug discovery pipeline to then predict which molecules need to be synthesized and used to combat something like COVID, so a disease like COVID. So this project is a virtual screening project, and I've already mentioned that Simon was involved, and a number of people from Galaxy EU and from the Diamond facility at Imperial. And so you can read through this. Here's the link. But effectively, what was trying to be solved there, as I've mentioned already, is taking some fragment hits, which is from a experimental technique, and then using a computational pipeline to generate targets that could be synthesized. So the pipeline then is as follows. There's all these hits that have been generated from experimental techniques, and there's a database of very many molecules, so 64 million. I'm not sure off the top of my head if that includes multiple poses, but I think not at this point. So enumerating all of those compounds, and then, as you know, for a docking process, there's a lot of things that you should do in terms of preparing your ligand and in terms of preparing your protein. So in terms of the ligand preparation, this section over here, that includes thinking about, well, what pH are we at, and do we want to think about the pKa of particular parts of this ligand that we are looking at, and enumerate all the charges that are possible. And you can also think of tautomers. The other thing is, if the molecules are given to you as a 2D structure or a SMILES format, maybe, how can you go from this graph format or line format to a 3D format which has Cartesian coordinates, because we're trying to dock this, as I mentioned earlier, this key, the ligand is the key, into the receptor or the lock, which is the protein. So we would have to generate 3D structures. And there's some other steps that you could put in place here. And then in terms of the preparation of the receptor or the protein, it's often the case that you want to remove any extraneous items that are in the protein structure that you would have probably downloaded from the protein data bank. And if not from there, maybe that's prepared in some other exciting way. So there's other tools that you could use to get to the stage. And then again, thinking about the pH, you would protonate the protein as appropriate, and you might optimize not just for pH, you might think about hydrogen bonding and all these kind of interesting things inside of the binding site. Then there's probably, hopefully, and in this case it was fairly well known, but hopefully you have a defined active site or cavity, and if it's not the case, then this becomes a bit more challenging, but often you know the cavity that you're looking to dock this multitude of ligands into. So you'll define that so that your docking software knows the available phase space it has to try out the simulation. So during the docking, in some ways, what you're then doing is you're taking all of these ligands, all of the enumerations of them, the tautomers, etc., and they have to have a 3D structure, and you're putting them into the available phase space you've defined as the binding site, and you're then calculating, you're going through a number of poses and calculating whether or not those are good poses based on an energy or a scoring function. So there's multiple scoring functions that you could use, and at the end of the day, what you hope to achieve then is a score or docked poses and also a score for all of these ligands. And then you'd hopefully then be able to top slice the best of these ligands in some way and use those to go forward to perhaps you actually want to go and synthesize or buy these molecules to conduct some screening later as well. So you want to top slice those. So in this case, the method for scoring was TransFS and sucrose, and then those were ranked. So you can see from fragments to ligand preparation to protein prep to multiple poses and ranking is this workflow that was conducted. And I think in the end, over 159,000 poses went through this workflow. So it was a lot of data that was analyzed. So with that in mind, you can probably imagine, according to what I spoke about earlier, that initially a history was created and the process was elaborated and checked, and at some point a screening workflow was generated. So this workflow is publicly available, and there's an all-in-one screening workflow generated by this team where there's the receptor, the active site details, all the ligands, and then docking and scoring takes place. So this actually contains multiple sub-workflows. This is the sort of simplified version of this workflow. But this is excellent because what it means is now that their work can be shared and double-checked and automated and all these great things. So I think this is the kind of thing that you want to do in a drug discovery pipeline. And Galaxy has already got a number of great examples, including this one. And you might find that you create new ways of running pipelines, and you can also do this kind of a thing. You can create these workflows and share them and automate them. So if you're interested in reading more about this, again, I'll mention that Simon will be giving the workshop later. So join him there, and maybe if there's time, ask about this work as well. But this is also freely available on the training service, and you can find out more there. And then just I think in terms of thinking about scaling up these large systems, where Galaxy is really useful is that when you've figured out your basic use case and you want to scale up, you can scale up through the web interface by using workflows and running multiple histories. But you can then think about empowering, maybe this is for your power users initially, but sort of empowering your users and taking them from just using the web interface to thinking about batch or automating their workflows. So if you're interested in doing that as well, this might be too soon for some of you. I don't know where you are in your drug discovery pipeline, but if you find that you want to automate, like I showed you all those histories earlier, that was done through automation, and it effectively means that you can write a single line on the command line using Planemo and run many histories. So if you're interested in doing that, this is a great training to have a look at. You can schedule and run tens or hundreds of Galaxy workflows easily. You can automate your analysis for large amounts of data. And what will happen then, once you've done that, you put all the work into the service, the Galaxy service. So the scheduling and the management all happens behind the scenes. And when it's ready, you can get an email. You will receive an email if you've signed up for that, and you can see the results of your analysis. So you can really power through these large data sets. So I think that'll probably be quite helpful for the future when you're building your pipeline up into a larger drug discovery pipeline. Okay, so just sort of the take-home message, I guess, for today is I wanted to share what is currently available in Galaxy and how that fits into a general drug discovery pipeline. So there's the chemical toolbox, which with a number of tools. We've got these workflows, you've got histories, and you've also got training and a great community that you can chat to about any of these tools, any of the histories, and also about bringing in new tools if you need those. So I hope that encourages you to get involved with your drug discovery pipeline using Galaxy. And then I guess one more take-home message is I really think what's exciting about Galaxy is anyone can do it. Anyone can use Galaxy. So you can do docking today. I mean, if you go home and if you have kids and they're excited to find out about what you learned today at this conference, at this workshop, you can say I learned about docking, and it's literally something that they can go and try out themselves. This resource is available for anybody. So it really then links into this idea of democratization of research resources and data. So you can run this from your phone, you can share your research with anyone. And we have a number of tools that I think will be useful to you in your drug discovery journey. So just, yeah, I'd just like to say in isiXhosa, which is one of the languages here in South Africa, Inkosi kakhulu, which means thank you, and thank you very much. I'm really grateful for the invitation from the committee, and specifically Kieran, who I've spoken to quite a few times. Thank you very much, Kieran. I'm also really grateful to the collaborators for this project, and of course to compute and infrastructure support people and services, and of course research funding. And thank you so much for being here today and for listening. And I'm really, yeah, it's been great to be here so far. I've enjoyed some of the questions already. I'm looking forward to taking your questions. Thank you.
Thank you so much, Christopher, for this wonderful talk.
Welcome, Sagita.
Yeah, and yeah, there are some questions for you.
Okay.
We can go through those. So the first one from Abhishek Chattar, how can we get molecular dynamics simulation of a protein dataset in Galaxy server?
Okay, Abhishek. Yeah, no, great question. I think it's a great question. Thank you. So I'm not sure where you are with your process right now. My suggestion is to look at the training. And if you're going to the protein ligands training later with Simon, Simon will also be there to help out as well. We have training for molecular dynamics, and I think I showed it in one of my slides. Maybe I'll send a link in a moment as well for MD training. So give me a second, and I'll share that link with you. And that will explain the whole process of setting up your system and running MD. Just to maybe expand on my answer, but if you have already got some system that you've set up outside of Galaxy, maybe the answer is a bit more complex. So that might depend on exactly which software that you've used. Right now, we support GROMACS very easily. And if you haven't set that up in GROMACS, that could be an interesting discussion we need to have. So I hope I've helped answer that. Maybe if I haven't and you want to ask a few more questions, you're most welcome to do so in the chat. And I'll send that link in a second.
Yeah.
Okay.
So shall we go for the next one?
Yes, that would be great.
Yeah. So this is from Dr. Jayaraman. How flexible is the workflow tools with respect to extracting information at some intermediate stage?
Okay, so I've just pasted that link that I mentioned,
Okay,
and then
sure.
how flexible is workflow with respect
Sure.
to extracting information? Okay, so yeah, that's a really great question. I think what happens when you're running a workflow, you might recall, and I guess I could show this again if needed, but you might recall when you saw some of the histories that I showed you, they were color coded, green and orange, and maybe there were a few red ones and gray. And gray usually means that the job is not yet, the job is not yet running. Green is it has run, and orange is, or yellow is it's running, and red is it failed. And so any job within a workflow that is completed, in other words, is green, you can see the full results for that. And if a job is in the sort of orange state, a lot of the time you can get some partial output for that. So I hope that helps answer the question. So if any part of the workflow is complete, any particular sub-job of the workflow is complete, you can get all the full details from that. And in fact, you could initiate another calculation on that if you wanted to. If the job is still running, it usually is accessible through the view interface. You can usually get a snapshot of it, but it's not always the case. So I hope that helps. I hope that helps you.
Yeah. So next one is from the INBIX team. In addition to Divya's question, what I would like to ask is nanoparticle docking with macromolecules is also possible using Galaxy. Because parameters for nanoparticles are not easily available in the available traditional software.
Yeah. Okay, so that's the most difficult question so far. Thank you. Great question, and I think the answer here is I'm maybe going to answer this sort of at maybe a bit of a meta level, but what Galaxy is helping you do is use the software that you have, your open source software, and link it up in a way that enables you to share and run these workflows, right? So what is ideal is that the software that you're installing and that Galaxy uses, let's say it's GROMACS. Hopefully GROMACS already has the force fields that you need for the nanoparticle parameters. And if the answer is no, then it's sort of a more development question. So if there's a software that is open source that does solve this problem for you, then we could bring it into Galaxy. If these parameters need to be more readily available and they're not yet, and you know how to make that happen, and you can start that discussion to help get other people on board who've got technical skills that might be able to help solve this. You could maybe engage with us in that way. But I don't think, and maybe I'll be corrected by someone, but I don't think that Galaxy can solve things that aren't working outside of Galaxy. So it has to be a functional code first. Yeah. So I hope that helps, and maybe you can share some of the details and the tools, and maybe there's a way we can bring those into Galaxy if you've got things that you need brought in. We can talk about that. Yeah.
Sure. Next one, can we use multiple docking tools in a single workflow and do the ranking of conformations in the docking exercise? This is from Shivam Pandit.
Yeah, so Shivam, thanks for the question. Yes is the answer to that, but this is then I haven't seen someone do this just yet in Galaxy, but the answer is yes, because in your history you can do your first set of workflows, do your first set of docking, your second set of docking, your third set, and you can create this workflow effectively, right? But if you don't see the workflow there just yet, it'll be something that you could create and maybe share with other people. But the answer is definitely yes. You can create very complex workflows, or you could create several workflows and chain them together. So the example I showed you from the COVID work, where each of those tools were actually sub workflows. So there's lots of flexibility. Yeah, you could do that.
Okay. The next one from Kripa Drishti, except for PDB inputs, what other data types and file formats can be imported into the Galaxy portal for analysis?
Okay, great. Thanks for the question. So I think the answer is very many. As you might have seen from across multiple fields, you could be importing data that's not just for chemistry, but just for chemistry. Off the top of my head, I can think of a few. So PDB, your GROMACS, your .gro type of file, XTC files. I think TPR files, which are also related to using GROMACS, DCD files, which is a type of trajectory. I'm sure we support many more. So there's quite a few. And I think and probably I think SDF. So you could have a SDF format with some smiles in there, etc. There's quite a number. And maybe the generalization I could make is, if it's a chem informatics format, we can import it easily. And if not, let us know through the forums or through Getter, and maybe someone can help. And if it's a structure format, so like a PDB or a GRO, or if it's like what I'm going to call a molecular movie format, it's a trajectory, right? We can probably import that. So those are the three general types. Yes, you can import any of those. Yeah.
Okay. Next one from Shivam again. Running docking, MD simulations, virtual screening pipelines in Google Colab or in Galaxy, which is more reliable and effective for students who don't have a powerful PC system.
Okay, so that's a nice question. Thanks, Shivam. So so I think both of those services try to solve that problem in some way. Like, we don't all have these fancy laptops or GPUs or an HPC right next door to us, so we want to use cloud computing. I think what you might find with Google Colab is maybe you might find some more flexibility initially with Google Colab. You might enjoy that. But I think with Galaxy, I think you can run your Jupyter Lab inside Galaxy, so it gives you that great flexibility. But the other thing is with Galaxy, you can schedule a lot of jobs to be run. Let's say you want to run 1,000 pipelines of something, you can schedule that. And I don't know that Google Colab can do that right now. So you can schedule all of these jobs to be run. They will run when there are resources available. The data will be kept for you. So if you look at the Galaxy EU details for the data storage and all that, it's kept for you. You can share this with other people easily. So yeah, I think they both have a lot of good use cases, but I think just generally your Galaxy makes sense. And there's also this community of people that have used these tools and are continuing to use the tools. Yeah, so obviously I'm a bit biased, but I'll say Galaxy. Yeah.
Yeah. Anvesh Pandey is asking, I saw your paper on using Gromax for MD on Galaxy. Now suppose I prepare all the necessary files on my local computer and just want to submit production MD job on the portal. Is this allowed?
Yes, anything is allowed. So, well, not anything, but most things. Yeah, you're most welcome to do that. And in fact, this is a workflow that's been used by several people here as well. Sometimes you want to prepare your system using. You might be familiar with Charm GUI, which is a web interface to Charm and helps you set up your system. So you can use Galaxy to do your Gromax setup, or you can use alternative strategies. You're most welcome to do so. I think the only thing that is sometimes an issue there is just to check that your force field file is set up correctly. And if you're having any problems, just message us on the Getter channels, and hopefully we can help you. But yeah, you're very welcome to do that. It's definitely allowed. Yeah.
Okay. Next one is interesting. I think Divya is saying there is some paid version of Chromax generally, and since the Galaxy is freely accessible, will there be any discrepancies in the results? How validated are these results?
Yeah, so I hope the answer is there will not be discrepancies, and I believe the paid version is more to do with support than anything else. Yeah, so I suppose the one nice thing about the version on Galaxy, yeah, we are using the freely accessible open source version of the Gromax code, and the results, should be the same. You give the system, its structure file and parameters, and you should see this is reproducible and repeatable. And of course, it's quite easy to test. So you can many times repeat the same experiment on Galaxy if you want to, and just double check that you're getting the same kind of results. So I think the answer is yes. And if you find that that is not true, that looks like a good paper to write. Tell us about it and explain how we can improve. And I think that's the whole point of these kind of open science projects, is they're meant to be transparent, and we can improve if we find any problems. Yeah.
Okay. Next one from Esther. Can we also use NAMD other than Gromax in Galaxy? So
right. So thanks for the question. So I'm just doing some Googling on the side, because the reason that we were struggling with using NAMD on the public Galaxies at one point was to do with the choice to use CondaForge as the main package provider. So I'm going to sort of unfortunately say I'm not sure to that question. If you have a local Galaxy that you're managing, you can install any software you like on the back end. You can install paid for software, anything you like. But on these public Galaxies, we do tend to, there's the tendency to install open source software available through particular platforms. So I think the answer there is a maybe at the moment, and I don't think it's yet on Galaxy EU. And it's something that I think most people are interested in having. So maybe if you want to message about that or request that, that would be really helpful. Yeah, thanks for that question.
Yeah, so Chris, that's all I have in the chat window, all the questions. And I think somebody from the INBIX team had raised hand. Is there something you would like to ask, Dr. Chris? INBIX team?
Yeah, Dr. Chris, I have a question. So, like, you specifically mentioned that you had done a charge enumeration of the ligands.
Yes.
So, yeah, why was it done? Like, since we already do clean the ligands, and if there is any, so is that the same purpose or something else?
So I think in this case, these were fragments, and I think there was an interest in looking at like tautomers and things like that. So sometimes those might bind better, and I think that was the purpose there. The best person to ask, I think, would be Simon, and maybe we could actually just have a look at that GitHub page. But I think the main thing was looking at tautomers and just looking at any system where you could have altered protonation that might be interesting, based on the pH, right? So yeah, I hope that helps answer the question.
Yeah, thanks.
Yeah.
Okay, perfect. Thank you.
Yeah. I think that I just missed that question in the list. It was there in the list. Yeah.
No issues, ma'am.
Yeah. I just have a small question, Chris, myself.
Sure.
Are there any software available in this particular platform which allow you to, compute and visualize, create diagrams of the interactions, the different types of non-bounded interactions?
That is a good question. Off the top of my head, I don't know the answer to that. So I'm just going to see if we've got something under the protein ligand tutorial. I'm just having a quick look there. I don't know that we do at the moment, and maybe that's something that we should look into. Off the top of my head, I do recall there being some open source software that can do this. And I just want to see.
There are software.
Yeah, so we seemingly don't have that.
Okay.
And if I don't know if Simon's in the room yet, but yeah, that seems to be something that might be missing there. Yeah. So Simon, I'm not trying to pick on you. Sorry if you don't want to answer. But yeah, it seems like we don't have that tool at the moment, so that would be a great opportunity then, something to add to the existing toolkits. Yeah.
Yeah, that will be a great thing to have. I mean,
we don't have that tool, unfortunately. So we just have the VMD visualization software, but explicit tool for visualizing interactions. We haven't integrated yet.
Okay, cool. Thanks for answering, Simon, and hi. Yeah, so that's like a great opportunity, I think. I mean, maybe I mentioned the GitHub page for Galaxy Computational Chemistry, and I'm sure there would be a lot of interest in helping get that tool into Galaxy if you're interested in doing that. Yeah.
Yeah, sure. Thank you, Chris. Thanks a lot for this wonderful talk. I'm sure all the participants have.
Sorry to intervene, Dr. Sangeeta. We'll take a group photo.
Yes.
Sure.
Yes.
Sure.
Sure.
Thanks.
Yeah. So request everyone to please switch on your camera so that we can take a group picture. Yeah, please. It's a rare opportunity. I think we should be able to utilize that. Yeah.
Okay, we'll go.
Please smile.
Okay.
Yeah. Thumbs up. Yes, thank you. Oh, yeah, thank you.
Okay, great.
Dr. Sangeeta, could you please unmute yourself?
Yes. So it has been a wonderful session, Chris, and thank you very much for giving us such a lucid, introduction to these computational biology tools for docking and MD simulations in the Galaxy platform. Thanks a lot, and thanks for the patience of answering each and every question in such a thorough manner. Thank you so much.
You're most welcome. Thank you so much, Sangeeta, and to your team. Take care. Bye.
Thank you. So with that, I think we have come to the conclusion of this particular session, and I would like Team INBIX to take over. Thank you.
Thank you, Sangeeta, for your wonderful sharing session. Yeah. So, without any further delay, now
we'll just take a minute so that we stop the recording and then restart the recording. Just.
Okay, Kiran. Yeah, please. Simon, just a couple of minutes, couple of seconds, please. Just come down.
I started the recording, but it's taking a bit time, just another 30 seconds.
Yeah, thank you. Okay.
So I just received a message of some technical issue on the Google Meet. So some of you might get will be gone out of the session, but you can log back in. So maybe we have to wait a minute. There's a technical issue.
Yeah, please be with us. Thank you.
Okay, seems to be fine. And if any of you got out of the, and of course I can't tell them because they are already gone right now. But yes, some of them, if now go out of this meeting, please come back. You should be able to come back again. So I give over the chair to the Sugunakar, and the speaker is Simon, who is a very active chemie informatician in Freiburg. Go on, Sugunakar.
Yeah, thank you so much, Kiran. And I welcome all the participants once again. And Simon, to this wonderful Galaxy workshop. To brief, Simon Bray is a member of European Galaxy team at University of Freiburg, and he's basically belongs to a bioinformatics group, Department of Computer Science at University of Freiburg in Germany. He's basically interested in computational chemistry, molecular dynamics, and the use of workflow management systems for virtual screening. He's been part of popular Galaxy chemical toolbox. He's finishing his PhD at the University of Freiburg, but has got extensive knowledge in protein ligand docking and cheminformatics. With these few words, I welcome Simon to take over the session. Yeah, thank you, Simon. Yeah, please, the stage is yours.
Okay, thank you very much for the introduction. So I will try and share my screen.
Yes, you can.
Yeah, please.
Let's see. Okay. And I assume that you can hear me okay? I don't need to make any adjustments to the microphone?
Yeah, audible enough.
Great.
Yeah, you're audible. Could you please a bit louder? Thank you.
A bit louder, sure. Is that better?
Yeah, it's perfect.
Okay, great. Okay, so then I guess I can start. So first of all, thank you very much for the invitation to present this workflow about protein ligand docking. So as was already said, my name is Simon Bray. I'm a member of the European Galaxy team at the University of Freiburg. You already heard a lot about the various tools and workflows for computational chemistry in Galaxy from Chris, and the idea of this part is to give you some kind of practical experience of how you can do these kind of analyses yourselves in the Galaxy platform. So I guess most of you have some idea about what this technique is. It's a molecular, it's a modeling technique for predicting the binding position of small molecules or ligands within a protein binding site. So the idea is that you have a particular protein which is of interest that you want to study, perhaps because it's implicated in some condition, and you would like to find some small compounds which can bind to it effectively and therefore change the behavior of the protein and therefore target the condition. So the idea behind this protein ligand behind this this technique is that you have a list of compounds, a compound library, and then for each of these compounds, you predict the best possible binding position of the ligand within the protein binding site. And this procedure is not something which can take place by itself. So
Simon, we lost you. You still hear us? Looks like he has a technical issue.
Simon Bray, are you there?
Okay, I will try to contact him. Okay, you can hear us.
Yeah, please.
Probably you might need to rejoin Simon, or I don't know if you have an issue. We can't hear you as of now. That's what the status. Okay, let's hope he will be rejoining.
We've answered for a trial if they want to do any transition. If they want to do, they can write it. Now most of the people are only problem is sharing it. There's no
Yeah, please, Simon. Yeah, please go. Simon, sorry, we are not able to listen. Just
now?
yeah, it's fine.
Okay. Yeah, so you might want to continue on to some additional steps, so some MD simulations or some free energy calculations. But the idea of this tutorial is just to do some basic protein ligand docking to give you some idea of the basic tools which are available in Galaxy for cheminformatics and computational chemistry. So I guess in the last that today, in the course of the various workshops that you heard and presentations, you already heard a lot about Galaxy, so I don't want to repeat too much here. But one point which I would like to emphasize is that it's a workflow management system. So it allows you to build quite complex workflows within this graphical interface, and then to run them at a high throughput level. So you can run them also on the command line once you have a well-developed workflow which you trust and you're happy to run on a library of 100 or even 1,000 compounds. And we have this specialized web server, cheminformatics.usegalaxy.eu. So yes, you can see the screenshot here. Contains almost 100 different tools for computational chemistry and cheminformatics analysis. There's also quite a range of different tutorials on the websites of the Galaxy Training Network, which I guess that you saw already. Today, we will do a relatively simple tutorial, but if you are interested in doing a more advanced topic, then we also have a video tutorial for high throughput molecular dynamics uploaded onto YouTube. If you want to go through this perhaps later on, and get to know some more advanced tools and some more advanced concepts in Galaxy, like how to run workflows from the command line, how to organize data into Galaxy collections as well. And the system that we will be looking at today is the N-terminal domain of the heat shock protein 90. So this is a chaperone protein, a protein which is sitting in the fold.
Simon, are you there?
Simon, we can't hear you. Okay, I will try to reach him out if there is any problem.
Yeah, please bear with us because of some technical issues. We'll be coming back soon.
Yeah,
thank you.
So I should be back. I should be back.
Yeah, thank you. Thanks so much.
Yeah, sorry about this. I just had some connection issues. So yes, I hope that you heard that I was talking about the protein that we'll be looking at during this tutorial. So the N-terminal domain of the heat shock protein of this heat shock protein. It's a chaperone protein which is implicated in some kind of cancer, and the idea is that if you can somehow block the activity of this protein, then you can slow down the process of protein synthesis, and therefore also slow down the process of cell growth. But the really important thing from our point of view is it's a simple globular protein which has a clearly defined binding site. So you have this beta sheet structure in the back, these alpha helices, between which you have this binding site where you have an example of a duct ligand shown in this diagram. And if you want to read more about it, then there's quite a nice review paper, quite old, from 2006, but still a good introduction to the HSP90 structure. Yeah, so that was all I wanted to say in terms of the introduction. Now I will go to the Use Galaxy EU platform and show you some of the tools which so please feel free if you're able to log on to usegalaxy.eu yourself. I think you have a link for joining the training, and you can follow along the steps as I demonstrate them. So I'm not sure if you saw this training icon already, but you can click on it, and it brings up this window in the center of the page. So I have it already on this protein ligand docking page, but if you're on this page here, then you can click onto computational chemistry and then onto the link here. Yes, so there's some introductory text and a little bit of information about the protein, which I already told you a bit about, that it's a chaperone protein responsible for helping with protein folding. And so the first step is to download a protein structure. So I guess that you know already that probably the most common format still for recording protein structures is the PDB format, and there's the protein data bank where protein structures are recorded. We have a tool also in Galaxy which allows you to download structures from the PDB based on their accession code. And so in this case, this is 2BRC, the accession code, which is associated with a particular crystallographic experiment that was done on the HSP90 protein. And so to download this protein, then you can just click on this Get PDB icon. And this is a very, very simple tool. I guess you used tools which are a lot more complicated so far today, and you just enter in the accession codes, in this case 2BRC, and start the job. So yes, I'll give my history also a better name. And we wait for it to load in our history. Okay, so now the job starts to run, and we can think already about the next steps. In this PDB file, then you have various different structures. So you have the protein itself. You also have a small molecule. So this molecule here, which is bound in the center, and in fact, I can show you that with the NGL viewer, which is embedded into Galaxy. So to get to this page, you can click on this visualization icon here. You have these options here, and you click on NGL viewer, and it takes some time to load. But you see this view of the protein, so similar to what I showed you before with these beta strands at the reverse of the protein, and then these helices at the top, and in between you have this structure here. And what we want to do is we want to remove the ligand, or rather separate the protein and ligand into two separate files, so we can then dock some new compounds which we will select into the binding site of the protein. So for that, as is described in the training material, we use this grep tool. So you can either click here, or you can search for grep in the tool panel, and it's this tool here. So search in text files, grep, and we first search for lines in this file which match the regular expression het atom or heteroatom, so the atoms of the ligand. I press that on execute. I run the same tool that once again, but this time I change match to don't match. So I have one file which contains all the atoms of the ligand, and one file which contains all the rest of the atoms, so the atoms of the protein. Oh wait, so actually this first example was a mistake, because the heteroatoms will also include the water molecules. We should instead search for the regular expression CT5, which refers to the code which the ligand is referred to in the structure. So yes, so I'll just remove that data set and then try once again, like this. So I have, yeah, so I hope that wasn't too confusing, but I have this first data set here, which will contain the coordinates of the protein in PDB format. So it will include all of the atoms which are not labelled as heteroatoms, and this file will then contain the coordinates of the PDB. Exactly. Yeah, this is also not correct, because I need to use the original PDB file which I downloaded, and not this file. So yeah, I'll try this again. But then, so you see that in this ligand PDB file, we can also inspect the contents. That we have a list of all these lines in the file which are labelled which labelled het atom, and in this column you have CT5, which is the name of the ligand code. We can also view this in the VMD viewer, and you can see the structure of the ligand. So you have this resource null structure here, and these 4 different rings which are connected. So yes, I expect that was quite confusing to see, but it should be explained correctly in the tutorial. So in the first step, then select lines which don't match heteroatom, and then rename the dataset protein PDB. And then in the second step, select lines which match this CT5 ligand code. Then this will be the ligand PDB file. Exactly. I can also rename this protein PDB file in the meantime. Exactly. So now I can visualize also the protein PDB file, and you should see that the structure has now been cleaned. Has now been cleaned. So the ligand has gone from the binding site, and we can start the tutorial once we have our prepared data sets. so yes, maybe I'll stop now for a couple of minutes and wait for you guys to catch up if you were if you didn't reach this step so far.
So meanwhile, if you have any questions, also you can ask.
Please do raise the hand always.
Oh, yeah, thanks for posting the link. Also in the chat to the training. Yeah, so perhaps I'll just continue. There don't seem to be any questions, and I think it wasn't too complicated so far.
There seems to be probably a question. Binta, for this, you want to ask any question? Okay, yeah.
Yeah, there's a question like oligosaccharides and monomers is same. When we use it as a ligand.
Yeah, I don't fully understand. I'm afraid. Can you provide a bit more detail?
Yeah, Binta Varghese, like, could you please unmute yourself and talk? Yeah, we can talk. You can unmute and you can talk. Not a bit of doubts.
Okay. Yeah, I guess that I will continue.
Yeah, please go ahead, sir.
Yeah, I will do. Right. So then, so in this tutorial, then we want to look at this idea of preparing a compound library. So it's a list of chemical compounds that we then can dock into the protein binding site. And so to process the compound library, then we use a tool which allows us to download compounds from this Chembor database. And the first thing that we need to do is to convert our ligand PDB file into the so-called SMILES format. So we can use this compound conversion tool for that. So we take the ligand PDB as our input file, and as output we select SMILES format, and the rest of the options we can leave as the default options. So yes, I'm not sure how familiar you are with different file formats for representing chemical compounds. So already we saw this PDB format, which provides the three-dimensional structures or three-dimensional Cartesian coordinates of molecules, and the SMILES the SMILES format provides representation of molecules as strings. So it doesn't provide the three-dimensional coordinates themselves of the atoms, but just but it just shows as a kind of molecular graph how the atoms are connected together. So once this tool completes, then I guess it'll be easier to see that. So I'll rename this data set to ligand SMILES. And let's see. Exactly. So if I press on this view data icon, then This is what the smile string looks like. So you have different symbols which represent different atom types. So obviously, C is carbon, O is oxygen, N is nitrogen, so this is a way of representing as a single string the same the same compound that we saw here. And we need the smiles format for the next tool, which is this Chemical search tool. So search Chemical database for compounds which are similar to a smile string, and we can then select the smiles input type. So we could either enter the smiles directly into this form here, or we can select file and insert this data set which has just been created. Then what this tool will do is it will search the Chemical database for tools which are chemically similar to this compound here, which we've extracted from the protein data bank structure. And this Tanimoto cutoff score is a kind of similarity threshold. So a high score means that it will only return compounds which are similar, and then a lower score means that you will get a wider range of compounds which are less similar to the original compound. So I will set this Tanimoto score to 40. The rest of the options I will leave blank, and then start the tool. And then what this tool should do is it should take this smile string, which I showed you here, and it will search the Chemical database and return a list of compounds in the Chemical database which are chemically similar to the original smile string. Yes, so there are various options here. So you can filter also for approved drugs. This will probably result in a very low number of results being returned. You can filter for molecules which are considered to be biotherapeutic or natural products, or for this Lipinski rule of 5, which is a kind of empirical rule which tells you how drug-like a molecule is likely to be. So you can feel free also to experiment with entering different parameters in this tool interface and seeing what kind of results you can get. So are there any questions so far? Is there anything which I should re-explain or
Yeah. As of now, like 14 persons are trying to do along with you.
Yep.
And I think if anyone got any questions here, please you can feel free to raise your hand and then ask the questions to Simon. Yeah, please. I think you can continue, Simon. I
think I'll continue. So this tool has completed. So we have 13 molecules which are returned, which is not very many, but
sorry to intervene, Simon. There is one question.
Can
you specify active sites in Galaxy?
Yeah, exactly. So that's actually what we need to do for this docking. So we have our list of compounds. and we want to then dock them into a specific active site. So in maybe, I don't know, 10-15 minutes, and I'll get to that point in the tutorial and show you how you then specify this active site for the docking step. So yes, I'll discuss that I'll discuss that shortly. So we have this list of compounds, which is obviously quite small for a docking study, but this is just a tutorial, so it's fine. Yes, and then I will rename the data set. So something like compound library from Kemble. And then the next step is preparation of the input files for the docking. So we have to prepare both the receptor and the ligands in our compound library. So let's start with the receptor. So for this docking, we will use this program Vina, and what it requires is the protein structure to be in this PDB-QT file. So this is very similar to the PDB file, but it has one extra column with the Steiger charges of the atoms in the final column, which isn't present in the PDB file. So to select a PDB file, we just take the protein PDB, which we have here. So it's important to use this one and not the original PDB file, which we downloaded from the PDB, but this one which we have cleaned, so it doesn't contain any water molecules or any atoms from the ligand, which is bound, and then we can just start the tool. And we have to prepare also the compound library, so the ligands that we want to dock. And again, we use this compound conversion tool, and we convert from SMILES to the SDF format. So SDF is more similar to the PDB format, in that it contains not just the molecule represented as a graph, but it contains also the three-dimensional Cartesian coordinates of the ligand molecules. So you can just click on this output format parameter and type SDF. And then what is important also is to generate 3D coordinates here. So because these SMILE strings don't contain any information about the three-dimensional conformation of the molecule, it's important that we use this compound conversion tool to produce this information about the shape and the conformation that the ligand adopts in three-dimensional space. And the SMILES format also doesn't contain any information about hydrogen atoms, so about the hydration states for the individual atoms in the ligand. So we should add hydrogens which are appropriate for a particular pH, and then a good choice is 7.4. So a neutral pH, and then we can just click on execute to start the tool. And in the meantime, this PDB-QT file has been generated, so we can also So we can also then rename it to protein PDB-QT. Okay, so at this stage, once this tool completes, then we have our PDB-QT file, which we use for as input for the protein for the VENA tool, which will do which will perform the docking. And we have our compound library in SDF format. So then for each ligand we have a list of coordinates, and in the SDF file then you have these. This is the separator, which indicates the point at which separates each of the molecules in the file. We have in total then 13 of these molecules in our SDF file. And the one thing which remains is the point which was already asked about in the chat, about how we specify then the active site for the protein. And we have a tool here called calculate the box parameters for an orthodox VENA job. So what this does is it defines a kind of box, so a space in which VENA will attempt to perform the docking of the ligands into the binding site. And what we can do as a kind of trick in this case is that we have our original ligand, which is from the PDB, and we know that this is in the active site. This is often the case that you already have some crystallographic structure, so you know where the active site is. And therefore, we can use the position of this ligand to help us to find the center of this box that we want to define, where we dock the compounds into. So we select here our input ligand or pocket as this ligand PDB file. So for the x-axis, we define 5 for each the axes, so 5 for the x-axis, 5 for the y-axis, and 5 for the z-axis. And this defines the number of angstroms which the box will extend on each side of this input ligand. Then exhaustiveness is the number of times that the software will try and search this box to find these binding poses. And 8 is generally a good choice, but we want to save some computational resources here because this is just a tutorial, so we set exhaustiveness to 1, and random seed. It doesn't matter what you set it to, but I think 1 is a good choice here. We press execute to start the tool. And then you might say, well, and this is quite a nice tool if you know the position of some kind of compound which is in the protein structure, which you can use to define this box, this active site. So I saw already that this has just appeared as a question from Shiva in the chat. So I'm not going to show you this directly in the tutorial, but there's a section in here on how to find the binding site of a protein which doesn't contain a ligand. So we also have this tool in Galaxy, which is called fpocket. And what fpocket does is it searches the protein structure for pockets in the 3-dimensional structure which are likely to bind small small compounds. So you can choose what type of pocket to select. In this case, this is a good choice. And then you can select your protein PDB file. And then what will happen is when you run this tool, is it will produce the PDB file for each of the individual of the pockets which it finds in the protein structure. And this is described in the tutorial. That it produces then a list of these pockets. What you can do with this box parameter calculation tool is you can use not just a file containing the input ligand, but also the file for a protein pocket. So this allows you to be quite flexible. You don't need to know the ligand. You don't need to know the position of a ligand which is already bound, but the advantage is that it is a little bit more convenient, and it allows you to skip this step with the F pocket tool. So in the meantime, then this tool has completed. So this creates a very small file. It's a configuration file which tells the Vena docking software these parameters. So it tells it the center of this active site, which is searched during docking with X, Y, and Z coordinates, and also specifies then the size of this box in the 3 dimensions. So in the X direction, the Y direction, and in the Z direction, and then seed and exhaustlessness are set to 1. Exactly. So then at this point we have our 2 files which we can use for the docking. The protein receptor file as well as the list of compounds, and we have our configuration file. So I guess I will just pause again briefly and see if there are any questions, or if people need to spend some time to catch up, and then we continue and do the docking itself.
Yeah, thank you, Simon. There's a question from Shivam Pandit. What if we don't have a co-crystal ligand? Then how do we define the active site box?
Yeah, so I talked about that a little bit already. So then the idea would be to use this this F pocket tool, which can provide you with a pocket. And then there's another question as well about the for defining the active site of a protein for the generation of grid box. Is it reliable to consider the active sites which are predicted using the online tools and servers?
Yeah.
I guess I cannot give a definitive answer on this. In my opinion, then the best thing to do is to read the publication in which these servers were published, and to try and understand a little bit what their process is by which these active sites are being generated. And if this makes sense to you, then I guess you can trust it. And it makes sense also then to view the active site yourself and look if it is a kind of sensible size and shape for binding small molecules. So I would say that you can use these kind of software, but then also spends a bit of time to inform yourself and to check yourself if the results of the web server or the software make sense.
Yeah, please continue, Simon. Thank you.
sorry, I was muted, but yes, I'll continue. So the next step is to do this docking with this autodoc Vino tool. And then, so for the receptor, we choose this PDB-QT file. For ligands, we take our compound library in SDF format. Ligand protonation we already specified in the previous step, but we can leave it as 7.4. And we want to specify a config file, which is here. And then we can run the tool.
Well, a lot of participants are actually not giving trouble to the Galaxy server. Only the 14 are doing, and actually a couple of jobs are queued in. And if you all use it, it would have been much more bigger problem. We would have seen a bigger problem. We lost the opportunity of seeing a bigger problem.
Oh, that's a shame. Yeah, so this tool takes a bit longer. I mean, this is the real simulation or calculation in this step. I don't know if there are any other tools in the meantime. If there are any other questions, I mean, perhaps then I can also continue with this optional section and demonstrate some of these tools which we have for example, for the visualization of chemical structures. So you can click on this visualization, select your compound library as SDF, and let's say that we display the molecular weight under the molecule, and we can run the tool. So what this will do is just produce a simple figure which shows the all of the compounds in our library in a two-dimensional in a two-dimensional view. So it will look something like this effectively. And then we have some tools also for calculating fingerprints and clustering. Perhaps I can demonstrate these as well. So, yeah, so for this we need to remove this second column here, this smiles file. So the best thing to do is to copy this smile string here, click on upload data, and paste data. We can set the type to smiles, so smiles. Click on start. Just rename it smile string. And then we can concatenate this with our compound library file. So we have a list of now all the compounds, including the ligand which we extracted originally from the PDB file. Yeah, and in the meantime, then the docking has completed. I don't know if you saw already today, we learned about collections in Galaxy. So in your Galaxy history, which you see here, then you have, it's a kind of flat structure. So you just have a list of data sets. And what collections allow you to do is to introduce a kind of directory or folder structure in history. So you can click on this collection here, and it shows you a list of data sets which are contained within the collection. So you have then in this collection you have 13 different data sets, so one for each of the docked compounds, and then for each of them you have a different number of docked poses, which the software has successfully found. So, for example, if we look at this ligand 1, then it's the same compound in each case. So this chemical 178-130, and so the structure is the same, but the coordinates will be different in each case. And what the docking does is it produces this score for docking. So here minus 8.3, then the second one is minus 7.8, minus 7.7. So scores which are below 0 indicate that there is an affinity between the ligand and the protein. So it's like with free energy, I guess. That negative free energy indicates that at the equilibrium, then the protein and the ligand will bind. So the molecule which is first in the STF file is the one with the highest binding affinity in each of these cases. Yeah, it seems that the data sets which are started are kind of stuck. I see that there's another question about the best value to be considered for the hydrogen bond length between a protein and ligand interaction after docking. And again, this is, I guess, a bit subjective. I would say maybe 2.5 angstroms. But I think most software will allow you to specify this yourself, so you can also experiment a little bit. And then, of course, I guess you should not only consider the length or the distance between the hydrogen and between the hydrogen you're considering and the oxygen or nitrogen, but also the bond angle, but the bond angle, so a bond angle of maybe less than 30 degrees away from a straight line is probably a good idea, but the different, so yes, I guess that would be my kind of personal answer, so less than 30 degrees and less than 2.5 angstroms, but I think that there are lots of different ways to answer that question. I don't think that there's one single right answer. guess that will then just wait a little bit for people to catch up and see if there are any more if there are any more questions.
Yeah, please. Any questions? You can raise or you can unmute yourself and then ask the questions, please. This is one of the most detailed and lucid talk. So it's important, like you get many questions, many doubts, especially the 14 participants who are doing along with Simon. It will be highly appreciable, and then it will be great learning experience that you can ask questions while doing your work.
Yeah, I mean, it's fine also if there are no questions. If something occurs to you after the talk, then you can also feel free to send the email or post in one of the chats, which I think Chris mentioned in the previous in his previous talk. But of course, any questions now would also be great.
Yeah. Thank you so much for that. Yeah. There is one question, Simon, in the chat. What is accuracy and efficiency of blind protein ligand DACON? Simon, are you there? Simon?
Looks like we lost him again.
Okay. Yeah. I think we'll join in a few seconds.
Yes, once again, some problem with the connection. Sorry about that.
Yeah, please. There is one question, like what is the accuracy and efficiency of blind protein docking?
I don't fully understand the question or what's meant by blind protein docking. I mean, what I would say certainly is that docking scores are not that reliable in of themselves. So I would advise you, if you want to do some kind of docking studies, to that you use the docking as a way to produce hypotheses. So you view them as potential binding positions, but then you also use some different method for the validation of the poses. So you have some insight into whether they really correspond to some experimental real experimental possibility. So, for example, I think Chris talked a little bit about the fragment screening, which on the SARS-CoV-2 main protease, which we've been working on. And there, what we are doing is we are taking compounds which are based on a set of input fragments, so experimental fragments, and then we're docking these compounds. And once we have the docked once we have these docked poses, then we are then comparing them with the original experimental fragments. So we can say that if the docked pose, which we get as an output, has some similar three-dimensional position to one of the input fragments, then we can say with some confidence that this pose is likely to be meaningful or have some or that we can rely on it. But yeah, I mean, I would certainly agree that just to do this docking by itself and to take the first one or two poses blindly is not likely to be a very good approach or very efficient approach. Okay. So in the meantime, this visualization tool completed. So you can click on this view data icon and get maybe a little bit of insight into the structure of these compounds. So one thing which I notice straight away is that you have this resorcinal structure. So this 6-carbon ring with these two hydroxyl groups here. And this is a kind of motif, which is very common in compounds which bind to this HSP90 protein. And it was also present in the original ligand, which we got from the PDB file. So yeah, it's quite interesting to see that that this motif is present in our compound library. Then in the meantime, we have got our concatenated data sets of our compound library with the initial
Simon, there is one question in the chat. If you can able to, yeah, please. Simon, are you around? Okay.
Yeah, so again, some issue with the connection. So there was this question about checking the synergistic effects of two compounds by combining them in ChemSketch and the docking in Galaxy. I think that makes sense as an approach, especially if you know that the two compounds have some activity or some particular functionality that you want to use, then you can like you wrote, you can try and combine them in ChemSketch and then to upload into Galaxy and to do this and to dock them into your protein of interest. Yeah, I think that makes sense as an approach. And then also would then say like in answer to the previous question is that you do this validation step where you then compare the results to the positions of the original compounds if you have some three-dimensional information about their position in the binding site as a way to do a validation of the results of the docking. So yes, I'll share my screen again. Yeah. So there's this concept of molecular fingerprinting, which is used to compare the chemical compounds and to judge how similar they are. The idea is that we have a compound with various different functional groups, and then we have a bit string, so a string with which is made up of zeros and ones, and then for each position in the string, if a particular feature is present, then we set it to 1, and otherwise we set it to 0. And I'll just go back to the questions. Yeah, so there was that second part of the question which I missed about the if there's any tool for predicting membrane and BBB permeability of the compound to trace release kinetics, and yeah, unfortunately we don't have such a tool. If you have some suggestion for software which can do that kind of prediction on the command line, then please feel free to suggest it, and perhaps at some point we can also integrate it into our platform. But yeah, at the moment, unfortunately, we don't have a tool for that. So yes, so in the end, for each of the molecules, we can calculate this kind of fingerprint. It will not work exactly like this with these 3 examples of functional groups, but in principle, then you have it works like this. Then you have various features in the molecule, and the bit string encodes the presence or absence of these features. And the advantage of then extracting this information is that you can then perform some kind of clustering, and you can then judge the similarity or lack of similarity between your compounds. So we use the molecule to fingerprint tool for this calculation. Select 27. And there are different kinds of fingerprints in this case. So I'm not sure which is recommended in the tutorial. Okay, so just the default these FP2 fingerprints. So let's start the tool. And then what it will do is that for each of these compounds, then it will calculate a fingerprint that which we can then use for molecular clustering and for calculating the similarity of our compounds to the original lead compound. Yeah, exactly. To cluster them and compare them to the lead compound. So if we view the fingerprint file, then it looks something like this. So it doesn't look like anything which is easy to read or to understand. But that's okay. So what you can see is that on these rows you have the chemical identifier from the chemical database, and then this is the fingerprint, which is encoded as a long numeric string. And then we can perform some different kinds of clustering. So there is this there is this Taylor-Butina clustering, which is used to cluster these molecules. So let's run this with a threshold of 0.8. Okay, so the clustering is completed. We then need to use this second clustering tool to actually provide some kind of visual depiction. So we can use this set here. Okay, on the threshold we can leave at 0.0. The format doesn't matter, but SVG is a better choice than PNG. And let's produce both of these outputs, I think. Then what we will get is some kind of plot as a dendrogram, which shows a hierarchical clustering of all the compounds in the compound library. Yeah, so hopefully this tool won't take too long. Are there any questions in the meantime? Anything which I should explain in more detail, or yeah, any general questions about molecular simulation or okay, it seems that the job has failed, so I will just try and rerun it. So normally if you encounter a failure, then you can click on this view details icon here. And then it produces some demo information about the tool. And then often it contains then these two rows, which contain the standard output and the standard error of the tool. But in this case, these are both empty, so I don't really have any clear idea about why the tool failed. So I'm hoping it's just some transient error, and when I rerun, then it will just then it will succeed this time. So let's see if that's the case. Okay. Yeah, so this time then the error is looks a bit more meaningful. Hairlines contain an equal sign. Yeah, no, I see what the problem is. I should have used, I've used the wrong data set as input for the tool. So I need to use this data set 28, which is which is FPS file, so the fingerprints, which I've generated from my compound library. So I'll try and run the tool again. I can remove these files. So hopefully it should succeed this time. Okay, this time it looks a bit better. So, I mean, that doesn't look so promising. Perhaps I should have set a different threshold. No, I'm not sure this doesn't really make sense. So what you have here is you have a similarity matrix. So we have 13 compounds. So we have therefore a matrix with 13 rows and 13 columns, and then each point in the matrix tells you how similar the compounds are according to this fingerprint measure. And for some reason there's some problem with the plotting of this dendrogram. So yeah, I'm not sure what is causing that, to be honest. But effectively you should have some compound, something like this. So you have a dendrogram, which shows you, for example, that you have your original ligand here, and that it's quite similar to all of these compounds here with these chemical identifiers, and then these two here on the left-hand side are then very similar to each other, but much less similar to the ligand and to the rest of the compounds. And then there is this further investigation stage, which is optional, and I won't demonstrate that here, but you can play around with these tools and try specifying some different parameters as well. Yeah, so now I'll show you a little bit about post-processing. So we have this very useful tool, extract values from an SD file or an SDF file. So we have this file which was produced by this collection that was produced by the docking tool. So we can submit it to our SD file, and then we can produce and what it will do is it will go through all of the molecules in each of the SD files, and it will check all of the properties which are specified in those files, and then extract them as part of a convenient table. So you can then take these properties and you can make some plots or filter based on these properties. I think it makes sense to include the property name as a header of the table, and to include the molecule name. And you can leave this field blank if you want to extract all the properties, or you can extract specific properties by specifying a list of properties separated by comma. So I'll press execute to start the job. And so what happens is that if we have a collection here which we used as the input. So this Galaxy collection produced by the docking tool, and if we then run the tool on a collection, then it produces a new collection, and it runs the tool individually on each of the component data sets within the collection. So if we look now at this ligand 1, and then this is just one example of the output. So it shows that for this particular compound, that you have this this list of poses. So like we saw before, the top scoring pose has this score of -8.3, and then you also have these RMSD scores, which tell you how similar the poses are to the most highly scoring pose. So these two have an RMSD value of 0, because this is the most highly scoring pose, but the rest of them then have a different RMSD score because they are more different. Then what we can do is we can also convert our SD files, which we produced back into PDB format. So here we have to select as the molecular input file our dot ligands, and because PDB files, in this case we want to choose this option, split multi-molecular files into a collection. So each of these SDF files in this collection contains multiple molecules, and we'd like to create a collection which then contains a data set for each of these individual molecules. So what we actually end up with here is a collection of collections. So I hope this is not then too confusing. Yeah, perhaps this is a bad example. So I'll remove that example and just demonstrate on a single file. So if we click on compound conversion, then I can select just a single one of those files in that input collection. So I'll just select this ligand 1. I can drag to my input file and then convert to PDB. And again, it'd be good to generate hydrogens at pH 7.4. I can press on execute. So yes, I missed this split multi-molecule files into a collection, so I can press on execute again. And then this time it starts to generate this list collection. So what this will do is it will produce a list of PDB files for each of these docked poses for this ligand. And then we can view this pose in using the VMD visualization. And what in principle you can do is you can concatenate this file with the original structure for the protein, and then view the compound as part of a hypothetical protein ligand complex. Yes, so I think that brings the tutorial more or less to an end. I don't know if there are any more questions. I realise that I haven't used my entire time, but I'm happy to take any more questions from the viewers, or if there's anything which I should explain again, or yeah, please just let me know.
Yeah, any more questions, please? If there are no questions, I think, Simon, are we done? Anything else you're trying to show?
No, so then we're done from yeah, as far as the tutorial is concerned. Yeah.
Okay, if there are no further questions, I take this opportunity to thank Simon So detailed analysis on protein ligand docking, starting the workshop series like one, started with Simon with a lucid and clear step-by-step analysis, like starting from cheminformatics, what is protein ligand docking, then separating protein and ligand structures, then a kind of hands-on that stepwise was being followed to show, and we've even witnessed around 14 students are trying to do along with him. That shows his clarity of thought and the work that need to be performed. Then he further showed on creating the processing of the compound ligands, generating compound library, searching from ChemMBL database, then further moving to preparing files for docking, then docking that's been performed by using the compound libraries which have been used, further visualization of the compounds, then calculating the fingerprints and then clustering. And then looking at the endograms, I think there may be some technical issues there. That's the reason we could not able to see. And further we see on post-processing and plotting. So nice to interact with you, Simon. And any questions, like they'll try to contact you through your email. Yeah, thank you for your time. And before we leave, we just take a photograph, and I give turn on your audio, video, please, so that we can have a video. Yeah, video image. Yeah, please, everyone. Simon, it would be great if you can able to stop presenting so that we can have a snapshot. Yeah, if you can stop presenting. Yeah. Kiran, could you please take a snapshot on your
Sure. Yeah.
Please. Yeah, please, everyone, request you to kindly turn on your video. Yeah. Yeah, please go ahead. Shivam, like, yeah, everyone, please. Shivam Pandit, Veena Rawat, Lipsa. Yeah, Santosh. Yeah, you should come forward. It's an opportunity that you basically share to keep your photo. Yeah, I hope everyone turns on the video, please. Yeah. 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 Do a smile. Yeah. It gives a lot of positivity. Okay. Ending up with a smile. Yeah. So we are closing a session with this workshop. Yeah, Kiran, now it is over to you. Yeah. Thank you, Kiran. Yeah. Thank you, Simon, for your wonderful session. Yeah. Thank you so much.
Yeah, thank you.
Thanks, Simon. Okay.
I think tomorrow.
Yeah, exactly.
Tomorrow's.
Yeah. Yes. I just shared once the tomorrow schedule. So thank you for all being here for today. You had a taste of one complete step-by-step procedure. Tomorrow you would have 3 more talks. Most likely they would also be in terms of the last talk what we had today would be a kind of more of step-by-step procedure. They are a kind of workshops, so you could always try to go, and the training links are also provided in the link what we said. So in the BioClues link, you can actually visit that link and check out all the links, what were the trainings used by all the speakers today. And about the first workshop, where the Vipin also showed, we will try to get the presentation so that you can also try to do that. And you can try, and you can also ask questions in the Gitter that will stay there. The questions people can come and then they can answer also later. So if you are trying to ask in the Gitter, the questions will stay there, and hopefully most of the speakers have the access to there, so they will be answering if required. And for tomorrow's, it will be you will have starting with a workshop 3, and then workshop 4 and 5. So it will be 10 o'clock start, a bit late start tomorrow. So you will have a bit more sleep. And after the first talk, tomorrow there is a lunch. So this is for the tomorrow's session. If you have any questions still, we will stay until you all hear here about regarding the using of the Galaxy, other things. You can ask any other question also right now. If you don't have any questions, then that's the day. That's it for today. But yeah, if you have questions, you can stay back and ask also. That's also completely fine.
Yeah, please. That'd be really good. Please stay back and then ask your questions. You might be having a lot of questions. Any questions regarding INBIX and those kind of stuff, you can always stay back and I can ask your questions. Yeah, please. We are here for about 5 minutes. We're here. Yeah, please. If there are no further questions, yeah, thank you for attending. Yeah, see you tomorrow morning. Yeah, thank you, and all.